Translational Divide webinar. Thank you for bearing with us. I think that, transitioning to home is going to make this webinar a little bit harder than we had hoped for, but we will get through it together. A few housekeeping things. Nicole, next slide. We just did some tests with some of you, but if you do want to speak, this is how you unmute yourself. We can share the slide deck in the Slack too, so that everyone can, jump around in the slides as they're watching the screen. We do want this to have the vibe of a discussion, but we also want to stay on track with time as much as possible. So I think after each XO1 presentation or each presentation, we will take a question, maybe one, maybe two, during the talks or in between each talk, but then we save more time at the end at the end of the XO1 talks for questions for kind of all the investigators. We've built in 2 10-minute breaks to our timeline. Those might get a little bit shortened if we fall behind time, but we'll do our best to stay on track. And with that, I will hand it over to Nicole Vasilevsky, who you all have been in touch with and has been great at coordinating this whole thing.
Hi, everyone. I'm Nicole Vasilevsky, in case I haven't met you before, but I think I've emailed with most of you. Just a few logistics things. If you go to this link, bit.ly/fen-slides, you can get to this slide deck here. I should have sent you the link to the agenda, and I also created a bit.ly link for the agenda available here, bit.ly/fen-webinar. And at the bottom of the agenda, I'll just show you, you can get to the, oops, wrong thing. You can see the link to the slides. There's a link to the notes document here. And then I'm also going to show you quickly how to use our brainstorming doc. So if you have the link to the agenda, you should have the link to everything. And then also, if you have a link to the slide deck, you can get to the agenda and the notes document here. So I created a notes document that's KF webinar notes. And the notes document is just currently a blank Google Doc, and it'd be great if anybody wanted to write any notes, any questions in there. We'll try to capture things as we go along. It's just meant to be general note-taking, brainstorming kind of things in the notes doc. And you can also put questions in the chat in the WebEx. So if you have questions that come up, we'll check the chat and make sure we get your questions answered. I just want to thank all the organizers for putting together this webinar. It's been a bunch of people coming together to coordinate this. So yes, Valerie, and then also folks from the Kids First team, Adam Resnick, Deanne Taylor, and Allison Heath, and then folks from the Monarch Initiative, Moni Munez-Torres, and Melissa Handel. We've all come together to try to put together this webinar for you, and we hope that you find it informative. And the objectives of this webinar are twofold. One, to hear from the different XO1 investigators and from some folks from the Kids First and Monarch teams to learn about the importance of deep clinical phenotyping efforts. And then at the end, we hope to have time to brainstorm ways in which we can improve our current methods in collecting phenotyping information. So there's a link to our brainstorming. It's not even a doc. It's a cool little application called DotStorming, and that's available here. And we'll bring this link up again at the end of the talks, and so we can all go in and brainstorm. But let me just give you a little quick overview of the brainstorming application that we're using. It's called DotStorming. And if you just go to this link, this will take you to this application. And I think you just have to just add your name, and then you'll be added as a participant. And then if you want to participate in the brainstorming, you can add a card by clicking up here in the upper right. And then it'll bring up a field for you to enter in your idea of ways that we can improve phenotyping efforts in this project. And then if you don't want to create your own ideas, you can comment on, or in addition, you can comment on existing cards by clicking this little button on the lower left-hand corner. And then you can also vote on the ideas that you like. And it looks like you have 3 votes available. So you could vote on 1, 2, 3, or give 3 votes to one card, whatever kind of way you want to. So if you have ideas that come up as we are going through the presentations, please feel free to get in here and brainstorm, or we can work on this all together at the end. So from there, I'm going to hand it over to Melissa Handel. She's going to talk about the importance of phenotype data depth harmonization for translational discovery. And Valerie, you're going to make Melissa the presenter, right?
Do that.
It's the same slide deck, though. Do you want me to? I can do it. Okay. Let's see. I always forget on WebEx. Here was this one.
It's there you go.
Working?
It says viewing your screen. We don't see anything yet, though. Yep, no.
I was unable to get it to work in WebEx yesterday at a different NIH WebEx. So is it working now?
Yep.
Oh, okay, great. Okay, so present. Okay, so I just wanted to kind of set the stage for what I hope will be a really fruitful conversation. And again, thanks to Nicole and Valerie and Allison and everyone who helped plan this. Obviously, we would rather be in person.
Melissa, I think we've lost your sound.
We lost audio, yeah.
Lost it too.
I think she doesn't realize we are not listening.
Melissa, we lost your sound. She's definitely still talking.
I just texted her.
She's saying amazing things.
The sharing privileges so that she realizes something's wrong, maybe?
You want me to take it away from her?
Yeah, just to bring it to her attention so she realizes that we can't hear her.
I can hand it back to Nicole. Yeah, it may help to turn off your video if for anyone who does end up having issues. Okay, yeah, we can have Nicole run all the slides.
Yeah. Technology.
Melissa, maybe try turning off your video.
Oh, she left the meeting.
Yeah, she's probably trying to restart. She was having some issues with this computer yesterday trying to use other platforms.
Well, should we do this a little bit out of order and switch over to Adam if he's ready?
That sounds good.
Adam, are
You guys,
I'm happy to try and do that.
okay, I will.
Here's Melissa.
do you hear me now?
Yeah.
Okay, let's try to have one person run the slides so that that doesn't happen again, since we have so many switches coming up. Okay, so thank you so much. Sorry about that, everybody. So I wanted to kind of give some background about why it's really important in the context of translational research and clinical care, as well as more specifically in the context of data being deposited in and utilized within the Kids First platform. Why, creating interoperability and utility and greater capacity from our phenotypic data and some of the challenges associated with that. So I'm kind of setting the stage for hopefully the other speakers to indicate kind of their particulars about phenotypic data. And then at the end, really having a deeper think about how can we do better to make the phenotype data that you all are generating be more useful, both for searching and cohort selection, as well as for analytics and mechanism discovery. So just really briefly, You're all kind of familiar with this kind of learning health system context. But here I wanted to talk specifically about the nature of the relationships between terminologies and data models. So over on the left, we have patient encounters. And in this context, phenotypic features can be documented. We have problem lists. We have the outcomes of clinical tests, often encoded in LOINC, for example. These types of data or in free text can be extracted either manually or automatically into various clinical databases. They can get delivered to registries and a variety of other sources. And eventually, we create inference and we create, quote, medical knowledge. In the case of Kids First, what's relevant is the relationship between genotype and phenotype. And of course, there's many other data modalities that are important too, but today we're focusing largely on that. And then this information, this knowledge goes into clinical guidelines and expert decision support systems that then help inform the clinicians and other caregivers on how to best manage any given clinical case. And so this learning healthcare system depends on interoperability in how we model the data and the terminological content that is used to describe those features, those phenotypes in particular, across this whole cycle. Okay, the next slide, please. So just briefly, this is an ontology, the Human Phenotype Ontology, developed by the Monarch Initiative. It's a standardized terminology, but it's a little different than the average clinical terminology in that it's than one parent to any given term. But more importantly, it has formal logic descriptions that not only allow maintenance of the ontology and interoperability with model organisms, which I'm not going to spend too much time on just now, but the focus, that logical interoperability is really, and it's, unfortunately, I'm working at home, so we're going to move to somewhere more quiet. That interoperability is indicated with some examples that where, for example, the term hyposmia is actually logically defined in terms of terms coming from other ontologies, such as the gene ontology, which might be annotated to other kinds of data. So, for example, the gene ontology term sensory perception of smell is annotated to 34,000. That function is annotated to 34,000 times in 22 species. So there's a whole lot of data out there that can be logically related to a phenotype observed in a clinical patient setting here if the data is encoded using an ontological term. And this is the same thing is true for other types of ontologically encoded terminologies, not just the HPO. And there's some links there if you want to, if you're not familiar and you want to explore. Okay, the next slide, please. So I wanted to talk about different types of phenotypic information. So I just kind of talked about what we call semantic phenotypes or sort of categorical phenotypes, descriptors using the ontological terms like hyposmia. But we can also have quantitative phenotypes. And here I'm using an example of, and also using it as an opportunity to talk a little bit about Fyler. So for example, Fyler has the Fast Healthcare Interoperability resource has what's called an observation resource, which is where we might record phenotypic features. Here, a PSA value on the left or a serum sodium value on the right. And each of these are essentially the outputs of tests that indicate a value that's in a range, and that range would have a reference range that would also be indicated, so you could know when something was abnormally high or abnormally low for the subject on which the test was taken. And those reference ranges can vary depending on the age of the person, the sex of the person, or even things such as ethnic background. Okay, so now we have quantitative phenotypes. Okay, next slide. These quantitative phenotypes can often be evaluated and using many different tests. So for example, here if we're trying to test for blood nitrite, that's an unusual circumstance to have nitrite in the blood, but there are many different
We lost you again.
Yeah, I don't think she can hear us. Nicole, maybe you want to write a note on the new slide and tell her we can't hear you.
That's a good idea.
Maybe she could call in. That'd be get the audio more reliably.
Yeah, that's one approach for her.
Is anybody texting her or?
I'm texting her on the side too.
Well, thank you, Moni.
How about now? Nope.
We can hear you now, Melissa.
We can?
We can. I couldn't hear her, Adam.
I thought you asked how about now.
I didn't hear that.
I didn't hear that either.
Oh, that was me. Sorry, that was Valerie reading her message.
I see. That's
Just give it to try by phone. So when she comes in, she's going to have to *6 to unmute herself, because it'll automatically mute her. Okay, I can send her the number, I think. Melissa, is that you? You might have to unmute yourself with *6. Should we switch to Adam and then I can try to send her the number in the meantime?
Yeah, that sounds good.
Yeah, I'm happy to take over.
Okay, sorry, Melissa.
You're gonna.
Yeah.
Yeah, I'm all right.
Yeah,
So.
Hey,
yeah,
Melissa's
there she
back.
is.
Can you hear me?
Oh, Melissa just got back. Okay.
Yeah.
I'm on my phone now. I think if you have low bandwidth, we're going to see this problem again.
Yeah.
Sorry about that, guys. Okay, so just really quickly, thank you for running the slides. multi device here. Can you hear me okay now? Okay, great. Okay, go on to the next slide, please. Okay. So I just wanted to, so this is really an example about tagging things. So there's also a lighter weight way to think about the many clinical data elements or columns that come in terms of the clinical data. So here, for example, we have many different variables that can all be tagged with a variable where it's really about HDL in blood. And so we can have a set of tags that people can search over if we curate those variables for those tags. And those tags could be ontology terms, such as from the HPO, or they could be just a lighter weight list of things that help people find the studies and the cohorts that they're interested in. So this is kind of a lighter weight way of sort of collating information from multiple studies or multiple cohorts using a tag for phenotype representation. Okay, the next slide.
Melissa,
So this is the last example I want. Yeah.
I'm sorry, quick question on that last one, if that's okay. But when you say something's tagged, is that tagged at the data set level, is what I'm imagining? This slide?
This is tagged at the variable level, but you would be able to subsume that set of tags at the data set level, right? So you could say, you know, out of these 1,000 columns, I'm going to tag, you know, these 20, you know, key variables, and then those would be key variables that have tags associated with this study or the data set.
Great, thank you.
You could also tag a study, but I think it's more informative to actually be able to tag the variable.
Great, thanks.
Okay, and this is a lot of what the TopNet group has been doing, and they've found it useful. It's a lighter weight approach, so it can be quicker, but it's less computationally useful. So it's kind of some of the trade-offs here. Okay, next slide. And this is related to the next example. So here, if you think about what many of those variables might actually be, some of them are coming from survey instruments or other kinds of case report forms. And here's an example where we were looking at anxiety measures. So on the bottom, we have an actual clinical data element that is generalized anxiety disorder 7-item questionnaire. Last 2 weeks, how often bothered by problem, feeling nervous, anxious, or on edge on a 4-point scale. Okay, so that's the question that's asked on that survey. On the right, from the same questionnaire, there's a summary total. So from some subset of questions that are on that survey, there's a derived score for generalized anxiety, of which that feeling nervous or anxious 4-point scale would be a part of. And so these are phenotypes too. So if you go to the next slide, and could conceivably be associated with things like our semantic phenotypes, such as the HPO term for anxiety, right? But we don't really have a systematic, structured way of actually doing that. And so we lack interoperability, not only from CDEs and survey instruments with semantic phenotypes and with some of the quantitative values, but all these different kinds of phenotypic modalities, and I haven't even gotten into imaging or histopathology types of phenotypes, which are also similarly challenging and need interoperability. So this is why we're challenged in the phenotypic space to really sort of make computable phenotype data coming from all the different data modalities and different types of studies. And so this is a charge to you all, to all the XO1s, but also to all of us, to really understand better how can Monarch and Kidsburst work with you all to try to make these phenotypic data more computationally useful, more interoperable for searching and cohort selection, and in the long run for mechanism discovery, treatment selection, prognosis, and other clinical care needs. So I think we go on to the next talk. Oh, I got a few more slides. This is just an example of like what you can do if you have that interoperability. Sorry, I'm a little flustered with all of my technology problems. So here is just an example if you have semantic phenotypes represented using the HPO. And so, you know, imagine that some of these phenotypic features might be derived from a survey instrument or from a laboratory test or from an imaging data set. Then you can actually use semantic similarity algorithms to compare patients against gold standards. So here in the middle is the Monarch gold standard for Wiedemann-Steiner syndrome, and these two patients were diagnosed with variations in the KMT2A gene based upon their similarity but non-exact match to that gold standard. And if you go to the next slide, there's one more. This is how you put it together with the phenotype data with the genotype data. So this is a very busy slide, and these slides are obviously available. But the idea is that we have genomic data, we have genomic reference data, and we can use a variety of frequency filtering, Mendelian filtering, pathogenicity determinations, and the like to identify candidate variants. But we really want to be able to combine these genotypic information with phenotypic information, coming from the patient phenotype data. And this is why we really, really need that interoperability, is because we want to be able to maximize the utility of that phenotypic information in the context of genomic interpretation by not only having the phenotypic data interoperability, but also interoperability with model organisms, with protein-protein interaction networks and pathway data, and many other kinds of omics data. So it's all about the context. Bringing that phenotype data is really the connector between all the omics data, clinical care, and the genomic interpretation. Okay, next slide. Okay, so on to Adam.
I'm handing you the screen right now. But you are still muted, so I don't know if you're talking yet.
All right, thanks so much, Melissa. This was great. Can you guys hear me okay?
Yep.
Can you hear me okay? Okay. So I think Melissa did an awesome job sort of setting the stage for today. And, from our perspective as a data resource center, we are really excited about today's program, in part because as amazing as Melissa's slides are, and as much kudos that she gives to our team, and especially working with her team, we have not yet solved some of the challenges. And so I want to take the opportunity to both show you where we are and where we're heading, and then set the stage for some of the brainstorm discussions in terms of how we can be even better. So the Kids First program is really unique in that it's bringing together not only two very distinct scientific realms, but also two oftentimes segregated communities. And those communities actually, as I think Melissa is hinting at, have differences in the way they think about data, the way they think about phenotypes, and the role that those terminologies actually have in their capacity to do research. And that's some of the challenges and the opportunities that we wanted to highlight. You will hear later from Phil Lupo, but this is, an all-time favorite Kids First program paper that, Phil and Sharon Fon, published recently, really substantiating this link between the risk factor for cancer or shared risk between cancer and birth defects. But, I think this was an enormous and an amazing body of work if we're trying to empower in the context of real-time research through platforms and interfaces. And so it really begs the question on how does the data come into the platform, what data comes into the platform, and how we can empower it. And we'll go through a couple of iterations on this. I'm actually speaking in a presentation today as a platform user. And some of these may or may not be universally true, these assessments, at least for me, but they're my interpretations of what I've seen our community engage in. And so for one, when we try to bring together cancer and non-cancer disease landscapes, what we face is really a tension, and the way that I characterize it sort of colloquially is that on the cancer side, the focus is on diagnosis, and clinical outcomes in ways that most of the data, non-genomic data on that side is, sort of on the mondo, quote unquote, side of structuring their data. Not Mondo as a whole, but in that format. Then at the other extreme, we have, I think really, the congenital birth defect researchers, who have, really spent an inordinate amount of time thinking about structuring phenotypes as part of the process of really integrating across the genomic landscape, for the disease. And then in between or straddling those two areas are really the syndromic context, oftentimes associated with chromosomal changes. So it's almost like, the chromosomal changes that have many genes in many contexts, have a human representation in the way that the community actually functions. But it's that tension that actually comes to bear when you start trying to figure out how to empower the phenotypic to genotypic connection, in part because of the source of the data and how that data comes into platforms. And so as a use case, what I wanted to just go through is sort of an experiment leveraging existing data resources that highlight the opportunity landscape between cancer and development in ways that actually at this point in time is not even using any of the genomic data within Kids First. So here what I've just done is taken two essentially lists, one from Cosmic and one is from Decipher. And so I'll just switch briefly just for those of you that might know one or the other. The Cosmic is a catalog of somatic mutations in cancer, and essentially, they publish consensus lists of what a curated list of essentially bona fide or thought to be bona fide cancer genes. Similarly, Decipher has a really dedicated effort towards the phenotypic landscape and the developmental disease context in ways that you can browse and download the data sets that are associated with particular genes and associated phenotypes. But if you take those two lists and just do a Venn diagram of that list, you'll see that a large portion of cancer genes, of curated cancer genes, overlap with the developmental disease context in ways that we can just do a small experiment on. So if we take that overlapping context of genomics and then try to do something similar in the context of HPO. So if I take and we extract all the current HPO terms from all of the Kids First data sets and look to see which of those HPO terms are currently overlapping with curated DDG2P gene lists, you'll see that there are about more than 400 of those genes have already been curated and associated with. That's my puppy in the background. I apologize. And then if you go a step further and look and take those HPO terms and ask the question, what have the developmental community associated in terms of genes with those HPO terms? And then take those genes and compare them across that intersection of cancer and development genes and just ask, within pediatric cancer cohorts that currently exist in Kids First, what do we see? So just as a convoluted sort of recap, if you take essentially the overlapping HPO term, the overlapping genes, do that intersection and query pediatric cancer cohorts within Kids First, what do you see? And this is just an example of if you just take the 100 genes that overlap between these contexts, and you'll see, right, that genes that are implicated in cancer that actually have mutations in relevant cohorts within Kids First are also implicated in developmental and congenital disorders. That was challenging for me to actually just describe, but it's even more challenging for a user to try and hop across these contexts and try to find and identify what these genes are and whether or not they're meaningful. And so that's sort of what we set as a challenge, to really undertake in thinking about the Kids First landscape and the phenotypes that can be intersected with genotypes. But a number of challenges then come to light besides this community-based structuring of data. The first is, where do data come from and what data comes in? So here I'm listing the HPO IDs, the number of distinct HPO IDs by Kids First study sets. You can see that there's a very wide range of HPO IDs. But what you can also see is that if you go to the diagnosis or Maunder terminology, the distribution changes. And again, you see a tilt towards, for example, the cancer landscape, right? So essentially patients that have a cancer diagnosis end up having many other Maunder terminologies or diagnoses associated with them. And that creates a challenge to link between these two, especially if data sets don't have both. And let me just go here to the actual portal and try to highlight for you this differences. So here we're actually within the KIDS referral within the Explore Your Data context. And if I put, for example, neuroblastoma as one of the data sets, you'll see that we have both kids' first neuroblastoma cohorts as well as target. And if I click either one of those and I apply, you'll see that we have almost 2,000 subjects included. But then in the portal, I'm going to try and see what observed HPO terms are associated with neuroblastoma. You'll see there are none. And again, that highlights the extreme as associated with standard ways that a cancer community approach might take to think about cancer landscape. And so this really limits the capacity to intersect in the phenotypic landscape. But if you take, let me show you an alternative example. If we'll take scoliosis, we'll hear today about scoliosis as a context, and I'll choose, scoliosis as an observed phenotype. You'll see that scoliosis begins to span, as you would expect, at least 4 different cohorts. And importantly, it spans 2 cancer cohorts. And those cancer cohorts are unique in that, in addition to the cancer diagnosis, there's been a clinical data pull from the electronic health record. It brings in, an initial, sometimes ICD-9 or ICD-10 code, but that, then a curation process has been undertaken to convert that to HPO. And it's only in that context, when you span those 2 domains of intersecting data that comes from electronic health records and data that comes from the research process of generating cohorts from specific disease communities, that you begin approaching this richness. But one of the other challenges, at least in thinking about how to empower these portal landscapes, is that HPO terminology is nested in the way that Melissa highlighted. And so this is actually an underrepresentation of that overlap, because it's really just looking for specific HPO terms. So for example, in this query here, you can see there's only really one proband patient, in the pediatric brain tumor atlas, and only one noted here. So I'm going to do something that Vincent's team did not expect me to do, but I'm just so excited about what Vincent's team has been working on, and they really pushed extremely hard to try and create a portal demo of some of the next features. You'll see in the slide deck there, they're actually screenshots because COVID and many other factors delayed the launch of this for a couple of weeks. But they've just done such an amazing job. I just want to see if it actually works while I'm doing it. So it's a risk space, but I think it's awesome. Let's do the same thing here.
I lost Adam.
Me too. Looks like his microphone's
Oh,
gone away. Oh, there we go.
I'm back on. I lost my phone connection, so I'm using my computer connection. So let's do the same search that we did here. So here we did scoliosis, and let's do it here with some of the new features that Vincent is bringing in. And we're doing this in part to show how we're trying to use the platform to help address some of the challenges as an end user. So if you go to the observed phenotype, here, instead of simply looking for a direct match, we're really trying to empower the ontological context. And if we start putting scoliosis, oops, it immediately recognizes that scoliosis is part of this very long branch, right, of HPO terminologies. And as I click on these buttons, what you can see is the relevant number of patients that fit into that. And again, from a biological context, as an investigator, I would want to start at the higher level, as high as I can, actually, to see if, maybe there are some, things that I should bring into view in ways that span the ontological spaces. So here, what you can see is that, suddenly, whereas a query here only brings you one patient in these 2 cohorts, when you really empower the ontological space, you start getting many more. So here you can see 131, patients across these, in the same exact cohort. If you have the capacity to really target and center around the phenotypic context and the way that it's captured. And this brings to bear the need to really think about how the medical system captures data, because different communities are going to capture different levels of specificity at the phenotypic level. And again, thinking about this differential context between diagnosis and phenotype as a mandate. But if you are able to capture that, you suddenly get really rich cohorts that can then really help you to prioritize those gene lists that are coming out of the genomic landscape. So I think, Vincent at St. Justine and the team did really, an amazing job at really thinking about these new tools, but there's much more to do, including implementation of some additional visualization that you'll see here on the left. But really, even beyond that, what I hope to get out of today's work is thinking about how it is that we can take the next steps to really empowering this data cohort. And let me just show you, the current landscape, if you just from a viewing of Maunder terms. So you can see, for example, here I'm just using, visualization of the number of studies that share a particular Maunder term, right? And you can see that there's, Maunder terms that have several studies that span them, but then it begins to drop off fairly rapidly. And these two studies that are in blue and dark blue are really cancer-focused studies that have integrated, a large amount of diagnosis. And so it begins to drop off fairly rapidly. Then you start getting into lists of just single Maunder terms. Similarly, on the HPO side, you can see there are a few, HPO terms that have broad representation, but then you begin to once again get restricted. And on the HPO side, what you start seeing is that it's really the studies that have deep phenotypes, like the PCGC, as you can see the red. Here you'll see pink, which is the brain tumor context, that are the ones that you'll see over and over, and then the orange. So you'll see that it's those studies that really allow you to dig into the phenotypic, genotypic context of those studies moving forward. So that's one of the challenges that I think we have in front of us. And really, this is something that Allison, I think, will talk about as she addresses Fyler and the next set of implementations that we should work through. So hopefully Vincent's team can breathe a sigh of relief, because I actually did try just the live demo and it was awesome. And you'll see these features that I highlighted coming into play very shortly, and we can further iterate them, but we have an opportunity today to brainstorm about how actually to further empower these platforms for that intersection of phenotype, genotype across cancer and congenital birth defects.
Thank you so much, Adam. I think we're going to keep things moving. So Nicole has control of the screen again, and Dr. Goldmus, you are next.
Can everyone hear me? I guess I'll get little notes if you can't.
Yes,
we can hear you.
Great. Thank you. So just let me say as a disclaimer, I'm not here necessarily representing PCGC per se. I'm here as somebody who has participated in reporting on their data structure and some other ideas around that. Adams and the previous speakers' concepts in mind, but hopefully this will be helpful. So next slide, please. Next slide. So everybody, I think, is familiar. Whoops, that's too many slides. Back one. Thank you. So CHD is, or congenital heart defects are the most common serious birth defect occurring in 1 in 200 live births and about 1 in 100 people, if not more. And the PCGC sort of published and stated goal is, quote, to identify genetic causes of human congenital heart disease and to relate genetic variants present in the congenital heart disease patient population to clinical outcomes. At the outset or through the past 10 years or 11 years of its existence, there have been more than 10 centers ascertaining cases. And in the first cycle of about 5 years, 10,000 cases were banked with their available parent samples and clinical data sets. And in the second cycle, an additional 3,000 cases have been banked. Next slide, please. So the PCGC data set is quite extensive, in fact. It includes basic demographics, a basic pedigree, which is really focused on birth defects, a pregnancy and birth history that's administered only to those cases less than a year of age, so that there was less chance of faulty recall. Details about the cardiac phenotype, which I'll focus on in a minute. We also include sort of review systems of other presenting congenital anomalies. There is a neurodevelopmental questions, I call it, that is administered to parents where the child is recruited at greater than a year of age. And I say questions because it's not an instrument, it's just questions such as, has your child ever been diagnosed with autism? And then a very limited medical history, detailing, I think, the first surgery if you were recruited, after that. But there has not been any follow-up, and so there's actually no clinical outcomes embedded in the PCGC data set, if you will. So moving on, next slide. There's huge challenges to coding something like congenital heart disease, as there are in any number of diseases. But first of all, the heart is actually an anatomically complex structure with hundreds of diagnoses, and I actually would call them diagnoses. There have actually historically been a number of different nomenclatures applied to this with some dueling camps over the years. And congenital heart disease can be described sometimes by a single sort of summary name, if you will, or set of features, or by its detailed component parts. And it's that tension sometimes of how much you try to summarize and bin versus how many detailed component parts you provide that can be challenging. In addition, the problem with it is there's a lot of overlap in terms of individual cases and how you might want to bin them if you're trying to give them sort of an overall diagnosis. Next slide, please. So, for example, I wanted to just show you a picture of a normal heart. You could call that a normal heart. It is a normal heart. But if you actually break it down into its component parts and you actually describe it, there can be abnormalities of every single component. So the veins coming in, the pulmonary veins coming in, the outflow tracts, the ventricles, the atria, the septi, etc. And even in describing the outflow tracts, for example, there might be subpulmonary, pulmonary, supravalvular, branch PA anomalies, for example, on the right side. So if you go to the next slide, if you look at that figure on the right-hand side here, I don't think you can see my thing. That is a picture of a simple diagnosis, tetralogy Fallot. Everybody would know exactly what that means, and it has certain component parts to it that you could describe. But if you just say tetralogy Fallot, that's its known, detail. On the other hand, that fails to describe whether that patient also had a concurrent secundum ASD, or concurrent venous anomaly, or concurrent aortic arch anomaly. And so again, it's this tension between giving single diagnosis or some of the more detail behind that. The thing that's easy is tetralogy Fallot is very well recognized, very well described. If you take something else like detransposition of great arteries, that can occur in many different contexts, either sort of simple or very complex. And you'll get a much bigger laundry list of possibilities that come to mind or diagnoses. Next slide, please. So one way of thinking about CHD and breaking it down is you have CHD overall on top, and then you have sort of these large groups of both by disease, by syndrome, by biology groups, but you can see that they intersect with each other, and this doesn't even represent all of them. Underneath each of these groups are individual, what I would call diagnoses. But you can also see that some of those diagnoses might belong under conotruncal defects, for example, like DORV and DTGA, or underneath heterotaxy syndrome, because you see it in that context very much at the bottom there, DORV, and TGA. And depending upon what you want to study, you might want to include DORV, TGA in the first conotruncal group, or you might want to exclude it. Or you might want to include those with heterotaxy, or you might want to exclude them. So there's, again, this tension between sort of grouping and splitting along primary diagnoses. Next slide, please. And then, of course, with any one patient, again, just featuring somebody fairly simple diagnosis of tetralogy Fallot, you would want to sort of detail each of the other features. So the way that we approached this for about, I don't know, 15, 20 years before the PCGC is we knew everybody had congenital heart disease, and we would force everybody into a primary diagnosis where they had to pick one. And this was our level of binning because it made it much easier later on to then assign them into a category based on the question you were potentially asking. And in addition to the primary diagnosis, which was a pick one only, we systematically detailed each of the findings for each of the sort of cardiac structures. And you had to give an answer so you knew that you knew it or you didn't know it going along. And that meant that you could either find tetralogy Fallot as a primary diagnosis with one click, or if you wanted to find everybody with a secundum ASD, you could go into the detailed the context in which something else was happening. Next slide, please. Next slide. So even so, we needed to create a hierarchy of priorities and make some arbitrary but at least consistent classifications. So for example, is a VSD coarctation listed under VSD or coarctation? And we listed them under VSD, which meant that coarctation was always a very clean data set of patients. And as I mentioned before, when you want to categorize them in even broader groups, you have to make certain decisions. So going on to what the PCGC did, which is a little bit of a hybrid. So the PCGC does provide first where the data was obtained from, so you can see what sort of the fidelity, if you will, or the detail of the phenotyping itself. Next slide, please. And then it took a little bit of a hybrid approach after much conversation. It does have the opportunity to say for each cardiac segment, normal, or abnormal, or unknown. But next slide, please. What it really uses to phenotype the patient are Fyler codes, which allows for very detailed description. It's sort of a laundry list of everything that is reported for that patient that you can find. And that's good for being comprehensive. But what wasn't, in my mind, perfect is that it doesn't require you to remark upon each segment, and you have to go, for time purposes, moving on to the next slide. This is an example, though, of what you end up pulling out. So if somebody has pulled everything in, then for each patient, you get this sort of laundry list of diagnoses. It's just everything. None of it is sort of hierarchical and not necessarily consistent. So for example, you can see that, I don't have a pointer, but tetralogy Fallot is the second person on the left, with far less reported than, say, tetralogy Fallot, the first person on the right, which has a lot more detail to it. And you don't know whether that was a difference in reporting, a difference in detail in the medical record, or whatever. And you can also see that if you get a long list of patients with all of this, you can miss it. I want to mention that the cardiac category to the left of the cardiac diagnoses all listed, that was assigned afterwards. So Martina Bruckner on her side for the exome data, and then my own group went through thousands and thousands of patients to actually categorize them as conotruncal or left-sided lesions. And in my mind, that was a fairly inefficient use. Next slide, please. Inefficient use of time. So my personal, questions about using Fyler codes or the way that we've done it, we have all the detail there, and that's really good. Or I'm not sure how consistent it was between groups. There was no categorization in that way, so you could search for everything, but you'd get everything. And it wasn't categorized by primary diagnosis or a superseding category that was assigned sort of later. Next slide, please. Again, trying to fly through this and hope this is relevant. I don't really know. I just wanted to mention that there's another coding system that has been developed. It was developed by the International Nomenclature Society, which met like 10 times in 40 hours and has been over 7 years, developed. Other clinical registries are using the IPCC codes or IPCC codes. It was done in conjunction with the World Health Organization for the 11th iteration of ICD codes. It provides very detailed definitions of the categories and diagnoses, providing sort of a tree structure all the way down to the single diagnosis. And it does consider every sort of component of the heart. It's, I think, really important for coding and outcome analyses, because if you want to compare, again, tetralogy of Fallot, you need to know that it's really the same patient phenotype that you're comparing to, say, mortality rates between centers, or complication rates between centers, to know that you're really doing it matched for clinical complexity. From a genetic point of view, from this sort of group, that may not be as important, because we're nowhere near understanding that kind of granularity, and I'm not sure it matters as much as saying somebody had TET, and they have TET, tetralogy of Fallot, and that's enough. So if you go to the next slide, I'm almost done here. This is a really tiny, it's really hard because it's a very long table now, and wide. But this is how the IPCCC is set up. You have these different levels on top, 0, 1, 2, 3, 4, that are sort of going further in detail, looking at the heart. And if you look at the middle segment, this is how you finally is an anomaly on the right side under level 2. All of those anomalies that are on the right side under level 3 are described. Then you hit tetralogy of Fallot, and under level 4 are the different types of tetralogy of Fallot based on the pulmonary valve. And if I could have taken more, it would just keep on riding down the list towards the right in its detail. In the IPC code is that they did provide really detailed definitions of the features of that heart disease. And that's in the first column there. And in the second column are sort of the lookalikes, or what other names tetralogy of Fallot, for example, might have been called. So in that sense, it's a great resource. I'm not sure it's the greatest way of coding things for the purposes of phenotypic data. But it is what will be apparent in the ICD-11 codes moving forward, and probably in other registries that will have more outcome-based data if we ever want to relate genotype to outcomes. And it provides strict definitions, which could be useful in trying to make data sets uniform. So my last slide. Again, I don't know if this is what people wanted to hear or if this is helpful, but thinking about integrating with other data sets, I think the importance is consistency, and providing a construct so that people can be consistent between what they're calling it and the level of detail. I think a summary diagnosis is actually really important and really helpful. It allows you, I think, providing the granularity that you can is useful, but doesn't necessarily need to be always so upfront. And obviously, all of this is limited by the availability of imaging records in the background of the reviewer. I would say this would be extremely difficult data to pull from an EHR right now. All of this in the PCGC, in our own studies, have been hand curated. And again, I think it'd be really hard. I'm trying to do some of that right now for other purposes, but I think it'd be hard to pull it from existing ICD codes. If they move to ICD-11 and they're using that IPCC coding, then that might be more doable. And I think that's everything I was going to say. I hope that was in time and useful.
Thank you. Moving on to Joaquin Espinosa. Joaquin, do you want just slides, or do you still want to try a demo? Joaquin,
Can you hear me now?
Yes, I can hear you. Do you want to try a demo, or should we just do your slides?
I'll do just 2 slides, 3 slides to give a little bit of background, then do that share screen trick that you showed me to do the quick demo. So hello, everybody. I hope everyone is staying safe and sane in these surreal times. So very quickly, I would like to give you a background of how we get to congenital heart disease and other comorbidities in Down syndrome as part of the Human Trisom project. So maybe we can leave it there at the project goals. The ultimate goal of the Human Trisom project is to enable a precision medicine approach to Down syndrome, a condition that is very variegated, very heterogeneous, with high propensity for many different comorbidities. Somehow, the extra copy of chromosome 21 causes a novel disease spectrum in this population, and they are predisposed to many, many things, including congenital heart disease. The ultimate goal is to enable the development of novel diagnostics and therapeutic tools that will benefit those with trisomy 21, but hopefully the typical population that may be affected by some of these conditions. Next slide, please. So the Trisom project, the goals are, of course, to accelerate the pace of Down syndrome research by completing a comprehensive panomics cohort study of trisomy 21 and create a large user-friendly public database for Down syndrome research. Next slide. So the title of my presentation, which we missed in the first slide, is phenotyping in the era of panomics studies and public research portals. And this slide is just to make the point that as many of us here in this call have been collecting DNA to do GWAS and other things, it is not that difficult from that same interaction with the research subject to obtain many other biospecimens that can catalyze the generation of other omics data sets. And again, I don't want to go through every detail in this slide, but it's possible from a single purple top EDTA tube to do panomics. Next slide, please. And then if you do panomics, it is also possible to bring all that data together in a relational system where then you can analyze that multidimensional data set by a number of strategies, like a public portal or what we call the deep blue team, the data scientists and bioinformaticians that can go very deep into that data, or even perhaps do crowdsource analysis with platforms such as the DREAM challenges. So we're big fans of these multidimensional data sets looking beyond the DNA. Next slide. And how do we do phenotype? We do online participant surveys, which can be provided immediately to the research participant. Some people fill this out very well, some don't, but we can always follow up on the phone and make sure that we understand the answers that are being provided. And we also do deep chart reviews. I should point out here, both of them have been modeled after what is the DS Connect survey. Is something that the community put together for Down syndrome, and we have built up on that and made it more sophisticated, if you will. And then all that goes into RedCap dictionaries that then can be fed into a SQL database with all the omics. And then on top of that SQL database, we can employ Shiny apps and set up a website to explore the multi-dimensional data set. I cannot emphasize enough that the key element here in this data ecosystem is the clinical coordinators looking at those medical records, at those participant surveys, and curating that data. Contradictions, the EMRs, even by the same doctor, sometimes by different doctors. Also, just see plain, misdiagnosis or and also a lot of a very different nomenclature. Is this a boil? Is it folliculitis? Is it hidradenitis suppurativa? Different doctors in different parts of the country calling the same thing different things. So with that being said, I'll like to show you what happens when we get this data and we try to make it talk to multi-omics data sets. And I'm going to share my screen now, if I can do that. I'm going to go. It's only two screens that I can. Great. Let me reload this. Can you see my browser, my Safari browser? Okay.
Yes, it's slowly loading, but yes.
I got disconnected from the browser, but you can all go now to this URL that is live. It's called trisomexplorer.org www.trisom.org/explorer. And this is live. Now, I'm looking at the internal version, the one that is not live, the one that we use here with the team, where we upload the data first, curate it, analyze it, make sure it's not full of bugs, and then we put it into the public version. But right now at home, you can play with this. You will have a couple fewer dashboards. You won't have the microbiome and the autoantibodies, but you will have all the other dashboards. So if I click here on clinical data, I'm going to get to something that looks like this, a little tutorial of how we got this clinical data, basically what I just told you. And you will see different tabs here on the left. We're seeing this cohort overview, condition stable, conditioning interactions. Got disconnected from the server. Valerie, you please tell me if you cannot see this. You can click on cohort overview. I'm gonna get an overview of the cohort. It's loading now. And I can see where we are today in terms of total number of participants consented, their sex distribution, age at enrollment, karyotype, ethnicity, geographical location, yada, yada. All of this is very interactive. I can look only, say, at the trisomy 21. So I'm going to click the control. The database now shows me only data on those with trisomy 21 and do only the males, that kind of thing. It interacts in real time. So coming back now to the issue of phenotyping for research and phenotyping for multi-omics. And I just love, by the way, the presentation that we just heard about all the various types of phenotyping congenital heart disease. We're going to see that in a moment here. We can go here to the conditions table, and you can see that as it was very well explained just now, you can categorize comorbidities at the level of a class of condition. For example, you can see here that people with Down syndrome have a lot more cardiac abnormalities. But then when you click into that, there's a whole lot of different types of cardiac abnormalities that our clinical coordinators can identify in the clinical trial. And I think many of these were just mentioned, right? And then again, you can do different groupings. You can do any type of congenital heart defect, or you can do any kind of atrial septal defect, or you can get very specific, very granular here. And then for the purposes of research and data visualization, you can start selecting different things. I'm just kind of randomly now picking up different things. And every time I select something by holding the command key, I get now an update of my figures. So I select these 4 conditions. 10% of people with Down syndrome, 11% almost, have at least one of those conditions. I can go down here and see my sex split for the 4 conditions, while I'm also seeing here the sex split for the major category. Now, what is very important in Down syndrome is that these conditions rarely happen on their own. There's always some of the condition going on. So I can visualize that here again at the level of class of conditions, specific conditions, by selecting the conditions that I want to look at. So I can go there to cardiac abnormalities and ask how that interacts with autoimmune conditions, how that interacts, say, with GI or neurological conditions. And in real time, I get a picture of what the cohort is looking like in terms of the various comorbidities. So yes, cardiac abnormalities are a lot more common in Down syndrome, actually the most common of the 4 conditions that I selected. And very frequently they occur alone. But as you can see, there's all kinds of possible combinations. So when we do GWAS or other omics, we need to keep track of these other combinatorial appearances of different combinations. And if I wanted to, I can go here to specific conditions. And now within heart, within cardiac abnormality, I start clicking things and see again the co-occurrence. And in real time, I hope this is all showing in your screens, I can see how various forms of congenital heart disease are co-occurring in the medical records. Okay, again, this was beautifully explained in the previous talk. So, so now coming back to how this impacts on the analysis of omics data sets, I'm going to just show you one data set, the proteome. I don't have time to show you much more than that, but I can come here and just ask simply what is the impact of having an extra copy of chromosome 21, and I can select proteomics, and this is mesoscale discovery assays, an assay to measure inflammatory markers in the blood, and I can look in this volcano plot proteins that are upregulating Down syndrome. There's a cytokine called IL-22 that is much elevated in Down syndrome. And I can zoom over and see TSLP is also elevated. I can come here and search for, say, TNF-alpha, and pointed then to TNF-alpha. And again, this is all, I can filter this. Now let me show you what happens when I want to ask within people with Down syndrome, what is the impact of having this or that comorbidity? So now I'm going to go here to effect of comorbidity, okay? And then I get to select my conditions. And we were in the business of heart disease. I can pick up, for example, that. Well, that's probably not exactly heart disease. Can pick up ASD. There's one more for the sake of demonstration. So now I selected 3 conditions, okay, here. So now I have cytokines that are upregulating those 3 conditions versus those that are downregulated. And there's a cytokine here called GM-CSF that I can click on. And it is upregulated in those that have the conditions that I selected. So then I can come back to my clinical data and say, well, I wonder if that was driven by aortic valve regurgitation or the other 2. So I can unclick that. My volcano plot is repopulated. And I can see that now GM-CSF is still up, but now I have another cytokine when I just have those 2 conditions, TARC, that is the winner here, the most upregulated. And of course, I can unclick one of the sexes. Now I lose power. My numbers have dropped significantly because I removed all the males. And now TARC is still significant, but the p-value has dropped significantly. And again, I can come back and take the 2 sexes and now ask only about heteros. Let me drop this farther to 18 years old. And now I have IL-8. In the kids, I have IL-8, PHIF. So this is how, if you have as an endpoint of the research, a Phenomics data set and a public portal, it really informs how you should do the phenotyping so that you can enable all these types of questions and interrogations in the context of an online portal where people are going to have all kinds of possible questions and inquiries, right? So I hope that demonstrates a little bit, how the clinical data interacts with the Phenomics data in the Human Trisom Project. Thank you, Valerie. That's all I have today.
Thank you. I'll hand it back to Nicole, and then Stephanie Sherman will present next.
Can you hear me okay?
Yes, I can hear you.
And Valerie, you're going to show the slides?
Yes, Nicole will share her screen.
Okay. Sorry, Nicole.
Okay.
So I'll continue the, in a sense, the use case of Down syndrome that Joaquin just presented very well. So my focus in this talk is on the complexities of harmonizing the neuropsychological data. So how did we get there? We studied Down syndrome and for the same goals that Joaquin just went through. And there's some 80 conditions and medical conditions that are increased among individuals with Down syndrome, and they're shown, by system in this diagram to the right. But we came into the Kids First program focusing on the congenital heart defects, and this came with both samples from the PCGC, from we just heard about, the phenotype data that are gathered from those, from Joaquin's Human Trisom project, and then from our own Down Syndrome 360 project. So where we've been collecting more than just congenital heart defects and really want to maximize the data that we get from the whole genome sequencing to more than just that one specific phenotype. So that got us to the neuropsychological data. And why the focus? Again, there's not just our data sets, but there's many existing cohorts in Down syndrome that have done specific neuropsychological test batteries on individuals with Down syndrome, but they're small samples. So that we need to try to find a way to maximize the data that exists. Very importantly, and I'm not saying this isn't true for every phenotype that we look at, but specifically, I would say for the neuropsychological data, there's a very high cost and a high cost to the family or the participant to obtain those data. And the goal here is to develop methods to try to automate the phenotyping so that it can be translated not just to Down syndrome, but to many of the intellectual disabilities disorders. And then lastly, I would say, at least for the Down syndrome community, and I would presume for many more, they really appreciate research that are moving towards interventions that might help increase the quality of life. And this is to at least to ameliorate the effect of trisomy 21 on the cognitive and behavioral aspects. So that's the motivation. And the next slide, please. And here is just an example of frustration when we tried to put a grant together to identify genetic variants on genes of cognition and behavior, knowing that they require very large sample sizes because these phenotypes, in a sense, I don't want to say they're weak, but they're messy. They're not a specific diagnosis. So trying to get large sample sizes with the best phenotyping data that we can is the goal. So we tried to put together a grant, 8 participating sites that had over 100 individuals with Down syndrome that were assessed by both survey and performance-based testing. They had in addition, the demographic and clinical data available. And next slide. And we couldn't do it. Our group ourselves didn't have the tools to harmonize these kind of data. We didn't have the actual resources to do that, and we didn't have the expertise. So what we ended up doing was disregarding all of our previous data sets and thought that the only way to do this was to create a new cohort. And that is just such a waste of investment into these studies, and again, to expand the burden on families who've been participating in these. But an ongoing, if you, next slide, the ongoing effort right now is to continue to categorize or catalog international cohorts to really understand what is out there in the existing cohorts. Can we harmonize those? And then can we use those data sets at the minimum to say how we need to start to develop a new cohort based on the information that we do gather with existing outcome variables? Can we do a better job at actually standardizing this effort? So that's an ongoing effort at this point. Next slide, please. And now I'm going to dig into many of the things that we've just talked about with respect to what Betsy was talking about with the congenital heart defects and how they to how they binned their data and also kept the granularity. So for any phenotype, I would say that the source of the data, the granularity, and the content of the data differ by each study. So by this, I mean, we can look at the least granular by parent report. Do they show symptoms of some specific behavioral disorder or cognitive disorder? We can gain more information by going into to the medical records and maybe interviewing with the clinician and their records to learn more about specifics and how they're being treated, by what medications, because that begins to again formulate the diagnosis. Next, we can go to parent report or teacher report psychometric testing, these surveys that provide essentially a screening mechanism for a particular disorder, a behavioral or neuropsychological disorder. And lastly, the gold standard, of course, is to go to performance-based testing and combine those with the parent report. So if we go back and think about something like autism, for example, parents can say yes, and maybe working with their educational system too, they can just say, yes, we have a diagnosis, yes, no, we have a diagnosis of autism spectrum disorder. We can go into the clinical reports again, that might be in there, it might not be in there, it depends if they're being treated by that specific medical doctor in that, or whether it's some other type of report that's not getting into the medical system. Is important to understand the context. The psychometric testing, I've just put, oh, next slide, please. If there's many different ways to screen for autism spectrum disorder. And importantly, I wanted to point out here, this depends on the age of the participant. So you gather information based on the specific time of the assessment, as well as these various surveys, and there's many more, these various types of instruments, ask specific questions that allow you just to get an indication of autism and perhaps other comorbid conditions. And next slide, which it shows the gold standard is to do an observational in performance-based test, the ADAS, combined with a very extensive parent interview, the ADI interview. So that can be done. It's very intensive, very expensive, and there's now other developing methods to try to get a more precise diagnosis of autism, for example, eye tracking, or combining the eye tracking with other aspects. So we've got the whole spectrum of the kind of data that we can collect to get at this one entity of autism spectrum disorder, for example. Could I have the next slide, please? I'm also giving an example of cognition, again, to emphasize that the components of cognition that we're interested in getting, because, for example, in Down syndrome, there's clearly strengths and clearly limitations in various domains, and we want to capture that in case there's different pathways that are involved in this alteration. But these measurements, of course, vary by age span. It varies by the developmental process and what we're asking of that child to do. So, for example, on the left side is an example scale, the Mullen scale of early learning, again, a performance-based test. And the scales you can see assess aspects of gross and fine motor function, as well as different components of language. If you look at the Kaufman brief intelligence test, this is one that's used quite often, the KBID-2. And this can be done in an older group. It has only 2 scales. It doesn't have a motor component. And then the outcome measures are something related to verbal IQ, a verbalized standardized score, and nonverbal. Both have a composite IQ score, and whether these could be used across studies is something to be understood. Next slide, please. So let me give you an example. So what I'm describing now is we, along with the really driven by the Kids First Data Resource Center, who are the experts at this, are trying to understand how best to do this type of harmonization. What can we pull from the various instruments, starting with our battery, but knowing that this battery is using standardized instruments that can be translated to other kinds of studies. It's really complex, and right now I'm just going to show you our thinking, though no results at this point. But how are we thinking about going from one, in this case, a parent report, to HPO terms? So the Connors is a good example. This is assessment of ADHD and also of other comorbid conditions that commonly occur with ADHD. It's, as I said, it's a parent observation about their child's behavior. It asks it asks 110 questions. They're all on a Likert scale, and they're all in the context of, in the past month, did your child, it can be applied to ages 6 to 18-year-olds. And it provides both raw scores and it provides standardized scores, T-scores, to help move from study to study. Next slide, thanks. So what is the content of this study? What are the kinds of measures that can be pulled out? There's an overall ADHD index score that can be used, for example, that might go across studies well. This would be the largest binning. This score is just a probability for this particular test where it's a probability of getting this score among a cohort of individuals with ADHD. There's also a global index score that expands that, and it's a T-score, and it's has two components: a restless impulsive plus an emotional liability. So that combines those types of behaviors. They also mapped the questions onto DSM-5 symptom scale. So this might be what we want to consider as what we pull from the Conners to make it more harmonized with other data sets if we base things on the DSM-5. You can see they split ADHD into predominantly inattentive and predominantly hyperactive impulsive presentations. They also have in their split out conduct disorder and oppositional defiant disorder. And so these are just based on counts of a subset of questions, and they're indicative of these different types of, or the higher scores are indicative of these types of symptoms, these DSM-5 symptoms. Or lastly, there's content scales. So you can see the list of different domains that are examined with these 110 questions. So again, subsets of these questions pertain to specific executive function, defiance, aggression, that type of thing. So what do we do with all these data? Can I have the next slide, please? Yes, so this slide just looks at the attempt of saying, can we map these onto the HBO terms for use? So the ADX or ADHD index score might best be mapped onto ADHD, yes, no. So if you get a score higher than some probability higher than some threshold, we can then map that onto that specific ADHD HBO term. When I looked at the symptom scales, at this point, there's no HBO term that begins to go to a more granular level of asking, is it is it just ADD or is it ADHD? And there's nothing for conduct disorder, but there is a term for oppositional defiant disorder. So which way to go, whether, we add these types of terms and would they make sense in terms of actually, would that help us identify genes specific to one or the other? And there's many, there's a large ADHD consortium that have started to answer that question. Next slide, please. And again, based on the content scales, are there HBO terms? And there's some that are similar to the domain. So inattention, there's an HBO term for short attention span. Again, hyperactivity, impulsivity, there's a HBO term for impulsivity. Whether these are getting at the same things is something to understand better. And it goes on with these peer relations. Again, there's two HBO terms: lack of peer relationships and impaired ability to form these. So again, the nuances of peer relations, but again, it could take you to a more refined assessment of a phenotype. Next slide. Yeah, so where are we now? I would say we went into this being very frustrated trying to figure out how do we dig through these data to even get started. And we had several starts and stops. And I think we're now at least at a point where we're more organized. And by we, I mean the experts from Kids First and ourselves who have the batteries and have applied these batteries and know Down syndrome. At least we have direction. So we are moving through it, and I think we have to go through this experience for these neuropsych data to really understand which direction to go. Next slide, just showing the final where we are headed. I learned very quickly that we need to work with the experts on these types of trying to move into the specific ontology. At first, we're even talking different languages. And so it's been a real learning curve for me and so helpful to try to say, okay, this is how we're using this information. Now, how do you harmonize it? So it takes both the experts from Kids First that are involved, and then those with that are working with individuals with Down syndrome. So of course, outcomes are the same as what we've heard. We want to increase the research capability. This is the first time effort for Down syndrome, so it's a huge stride forward. The Kids First DRC, are now, once we get again more clear about which direction to go, they can use some of the tools that they've already developed to initiate this effort for the neuropsychological data. And of course, these tools will be available not just for the Kids First projects, but for also the Trans NIH initiative. So we're very excited that this is moving on. And as I said, I'm very excited to be working with this team. It's a new concept for us in detail. So it's something that we've always wanted to be doing. Thank you.
Thank you, Stephanie. We are running more than 30 minutes behind, so we just need to keep this moving as much as we can. We will turn it over to Philip Lupo, and then we will shorten our break to 5 minutes. And then if the presenters who are presenting later can look at their slides and prune where possible, that would be very helpful in the meantime. I'll send the slide deck link right now. So Philip?
Thanks, Valerie. I'll, even though I'm going to be talking about 2 projects, I'll try to make up some time. So obviously, to start out the conversation, I'd like to remind everybody about cancer risk in children with birth defects. And as you can see from this really nice figure from Lorenzo Botto's paper, that risk is exceptionally high for children with underlying chromosomal anomalies. So if you look at the next panel, you can see that relative to children without Down syndrome, those with this underlying condition have about a 20-fold increased risk of developing acute lymphoblastic leukemia, which is particularly acute in early life. So next slide. Aside from an increased risk, children with Down syndrome that are diagnosed with ALL also have inferior outcomes, which is probably associated with the distinct immunophenotypic and cytogenetic characteristics that they have. So for instance, they have a higher proportion of CRLF2 overexpression and cooperating JAK2 activating mutations, and a lower proportion of these children have more favorable subtypes of leukemia like ETV6, RUNX1. But in spite of this predisposition, it's really unclear why some of these children develop ALL while others don't. So to that point, go to the next slide. Collaborators on this call and I conducted the genome-wide association study of Down syndrome acute leukemia. And you can see even with a relatively limited sample size of just over 500 cases, we were able to identify some relatively strong and significant associations. But these are variants that have been identified in GWAS of ALL independent of Down syndrome. So that really made us wonder, if there were other genetic factors that could be underlying this condition, especially on chromosome 21 or throughout the genome. So moving to your next slide, or to my next slide, that was really the underlying premise of our X01 application. And we actually bundled this work with Stephanie Sherman in the samples and data that she's collected on children with Down syndrome and congenital heart disease. And our specific goal, at least the genetic association analysis or genetic analysis, was focused on genetic variants underlying ALL in children with Down syndrome. So moving on. The samples included in the project will be 408 germline tumor pairs that have been obtained through the Children's Oncology Group. I just wanted to give a little snapshot since this is I'd say a younger project in terms of what's been sent for sequencing, which you can see here. Next slide. But I wanted to talk about primarily in this session and this meeting about a potential DS-ALL phenotype. And these are data from the registry study that Adam pointed to earlier. There were actually about 160 children with Down syndrome and ALL. And you can see compared to children without Down syndrome, or children with Down syndrome that did not develop ALL, there was actually a higher likelihood of those children having co-occurring birth defects in some of the organ systems that we think of as being affected in these children. So, for instance, the children that went on to develop ALL were more likely to have gastrointestinal defects. They were also more likely to have congenital heart disease. And then, as you can see at the bottom of this table on the left, they had a higher number of co-occurring birth defects than the children that did not develop acute lymphoblastic leukemia. So we took that threshold of 10 or more co-occurring birth defects as sort of a dichotomous definition of phenotypic severity. And again, you can see on this right panel that the children with 10 or more co-occurring birth defects were about 40% more likely to go on to develop acute lymphoblastic leukemia. So next slide. Unfortunately, and Stephanie and I have talked about this many times in thinking about pooling the data that we have through the Children's Oncology Group, there is no sort of pre-existing phenotypic data on the children that have Down syndrome acute lymphoblastic leukemia. And you know, this is primarily a function of the lack of bandwidth for collecting this information systematically on children with Down syndrome that develop ALL. So along with Karen Rabin and others, we've obtained preliminary funding from the Include project to rectify the situation. So our goal is part, or at least in conjunction with the sequencing efforts that are ongoing as part of our XO1, is to conduct deep phenotyping of children with Down syndrome ALL to identify congenital risk factors for DS-ALL, and also to see if there's an impact on clinical features as well as outcomes. So there's actually wonderful mechanisms available through COG to recontact these individuals. And then through that process, we can obtain medical records. And we're also planning to administer the same questionnaire that Stephanie has administered through her ongoing efforts in the DS360 study to at least ensure harmony in relation to some of the structural birth defects and other phenotype data that we're hoping to collect. And then aside from those data, we're hoping to create sort of a data warehouse of outcomes in these children as well. And that's going to be collecting any information from clinical trials that these children were enrolled on, which would include toxicities, as well as linking this information to the National Death Index to evaluate differences in long-term survival in children diagnosed with Down syndrome ALL. And next slide. So that's it on the Down syndrome ALL project. The next project I'm going to be talking about has nothing to do with Down syndrome ALL, but is an independent project that's been funded through the Kids First program, and that's on rhabdomyosarcoma. So next slide. Just for a bit of background, rhabdomyosarcoma is the most common soft tissue sarcoma in children. About 50% of cases are rhabdomyosarcoma. Notably, these tumors can occur anywhere in the body. There are two histologic subtypes that I want to talk about when we're thinking about phenotype, at least in relation to genetic susceptibility, and that's embryonal RMS or ERMS and alveolar RMS. And those respectively make up about 70 and 20% of cases. So next slide. Aside from histology, if we dig deeper into somatic characteristics based on histology, you can see that the alveolar subtype are largely characterized by these packed FOXO1 fusions, and they have relatively few somatic point mutations or driver mutations in cancer genes that we typically think of. However, if you look at an embryonal tumor, we see a lot more classic point mutations in some of the classic cancer genes that we think of for sarcomas and other pediatric tumors. And if you'll go to the next slide. In terms of genetic susceptibility, it's estimated that about 7 to 33% of these patients have a pathogenic variant and a cancer predisposition gene, but there's really been no large-scale effort to confirm these estimates. And there's also been no genome-wide association study or exome-wide evaluation of rhabdomyosarcoma. Next slide. And I just want to show you some of our preliminary data with the point of, again, thinking about how to better define a phenotype in relation to genetic susceptibility. So we curated a list of cancer predisposition genes and divided them into rhabdo-associated genes that have been at least reported in case studies or in clinical observations, and divided that group into a separate bin and evaluated other cancer predisposition genes, which you can see at the bottom of this table. So if you go to the next slide, you can see some of our preliminary results, which suggest that children with rhabdomyosarcoma are about 5 times more likely to have a pathogenic variant in a cancer predisposition gene than a group of unaffected controls. Not surprising. You know, if you do look at our curated list of rhabdo-associated genes that we identified, that relative risk increases to about 12 versus 2 if you look at other cancer predisposition genes. And then on the right panel, you can see the recurrent mutations that were found in a list of cancer predisposition genes. And the reason I wanted to bring this up in particular, you can see 5 of our patients out of 600 had a pathogenic variant in HRAS, which would be indicative of Costello syndrome. Again, this is the type of phenotypic information that isn't traditionally captured, at least systematically, in the COG data that we have. So going on to your next slide. This is sort of the meat of the study and thinking again about using our tumors to better phenotype. So about 7.8% of the patients had a pathogenic variant in one of the cancer predisposition genes. But what's interesting is that when you look at differences by histology, 10% of children with embryonal rhabdo had a pathogenic variant versus 3% with the alveolar subtype. And then if you further stratify in the alveolar cases where there was a PAX-FOXO1 fusion, absolutely none of those cases that had a FOXO1 fusion had a pathogenic variant in one of the cancer predisposition genes. So next slide. Just quickly looking to the right-hand side of the panel, our XO1 has really focused on 2 independent populations. One is a recruitment study that we have through COG that's going to be similar, or that is similar to the Down syndrome ALL study, where we've been recruiting throughout the United States, and we've recruited a little over 200 case parent trios to date. And we've also been relying on biobanking efforts in COG to obtain 600 germline tumor pairs. Next slide. So the phenotyping efforts for rhabdomyosarcoma, I think, are also going to be, or at least I hope, potentially rich for thinking about a lot of different questions. So in our trio-based study, we're collecting questionnaires and also medical records for these individuals, and that would include information on birth characteristics, exposures encountered during pregnancy and after for moms. The COG biobanking data can be linked to demographic data as well as tumor characteristics. We'll have outcome data on those that were enrolled in clinical trials, so that's going to include toxicities. And again, because we'll have the tumors, I think as we start generating the sequencing data on the tumors and can sort of categorize our cases into molecular subtypes, that's going to inform, or at least I would hypothesize better inform, our genetic susceptibility studies. And I think that's it. I do want to thank everybody. I'm really disappointed that we're not able to meet in person. I was really looking forward to this meeting, and I'm glad we can do it in this venue. But a lot of people on this call have been in this effort to some degree or another. And again, a big thanks to all of those individuals. With that, I'll end.
you so much, Philip. You know, since we are short on time, we're going to pause now for a 5-minute break. Let's come back at 2:50, 2:50. We may make some tweaks to the agenda, and we'll share that with you before we start at 2:50. Thank you, everyone. Give us one more minute and we'll be back, folks. Okay, we have just been trimming the afternoon or the remainder of the agenda as much as possible. I think the hardest thing for us is that we're really loving all of these presentations. They're all very valuable. So we don't want to cut into anyone's time. So we forfeited ourselves. We cut into our time. So Nicole, are you ready to share your screen again? Almost.
Yep, I'm ready.
Okay. And again, we did request that these presenters cut slides where they can. So now we'll turn it over to Nar Zabrera. Please, we'll go back. Sorry, Nicole. Take a moment to look at this revised agenda. This is on slide 84, if you want to reference it. And this is what we'll use to guide us through the rest of the meeting. Now turning it over to Nara.
Hi, can you hear me?
Loud and clear.
Okay. So I'm going to talk about the enchondromatosis project that is going on through the Kids First grant. And since we are talking about phenotype, I'm going to talk, the first slide is just to say that these two syndromes that I'm studying, Olea disease and Maffucci syndrome, are really not well known in terms of the phenotypic spectrum of the diseases. So we are not really sure of the complete phenotype of each one of them. So I have been working on trying to get information from the literature and from the patients themselves to understand better what the phenotypic features of these syndromes are. And as part of this effort, we just published last month the natural history of Olea disease and Maffucci syndrome based on a patient survey and on the review of clinical literature. So we sent an online patient survey with 90 questions that at the time when we put the paper together had been answered by 126 patients with Olea and 36 patients with Maffucci, and these patients were from 27 countries. But as of yesterday, it had been answered by 211 patients. And we reviewed 359 cases that had been described in the literature. And by reviewing these cases, I wish the journals had a template for gathering phenotype from these patients that was a unique template that every journal would use, and that when we are getting the phenotype from the patients in these articles, everything would be just like as standard and uniform as possible. But we all know that's not what happens, and it's really hard to get phenotype information from papers that are written each one in a different way. Next slide. So as part of the Gabriella Miller Kids First project, and also from the Baylor Hopkins Center for Mendelian Genomics, I am also part of this project funded by NIH. We have consented and collected phenotype information of 138 probands, and we have sequenced 105 of these probands, including 75 trios. So between the information that we are getting through the survey and the patients that we are collecting samples from for the sequencing project, we have a total of 239 patients that we have collected phenotype and or sequencing data. And where are we storing all this information? It's what we have been working on. Next slide. So as part of the Baylor Hopkins Center for Mendelian Genomics, 8 years ago, we created a database that's called PhenoDB, that is a phenotypic and genomic database. And all the information that I have been gathering from these patients, the phenotypic information mainly, it's in this database. And I'm just going to show a little bit of how it's organized in there. And like many things we have actually been changing throughout the years as we face new needs for the phenotyping and also for the storage of the genomic information of these patients and others. PhenoDB, the Baylor Hopkins Center for Mendelian Genomics, PhenoDB now has actually 8,811 patients in there. 4,128 are affected individuals. And if we look for the features that are describing these patients, in average, each affected individual has 4.4 abnormal features that are described. And in total, between abnormal and the submitter saying that it's not abnormal, they have 7.3 features per individual in there. So my cohort of enchondromatosis is in this database. And how I take advantage of the other information that is there is what I'm going to show you a little bit now. If you want to check PhenoDB, you can go to phenodb.org and see some of the features that I'm telling you today are in there and more. We don't have time to go over everything, of course. So next slide. In PhenoDB, we can put the features of the patients using the PhenoDB ontology, but it's all mapped to HPO, as you can see here, and also it's mapped to the ICHPT terms and to the elements of morphology terms. Next slide. So when I put the features of my patient in there, I generate a table like that. You can see that this patient has enchondromas and he also has hemangiomas, and I can download this table as an Excel file. So one of the things when we were actually giving the information to the Kids First project, there were things that were asked us that we have in the database, but are not in this table. And there are also information that we don't have in the database, or like the date of birth for the parents, I don't have it at all. And also the ethnicity, we have it, but we were asking in a different way. So now I'm trying to make it uniform and adapt this table in a way that when I export the table, I don't have to be modifying anything. I can just export it the way I need it to be already. But the other thing that you can do in the database is actually, next slide, it's fine. You can generate a table for many patients at once. So in this table, I actually selected 5 patients from my cohort, 5 different patients, and I get the information from the affected individuals in these families and from the unaffected individuals, all in one table where I can actually compare the patients across the entire cohort and see what they have in common and what they don't have in common, what are things that are unique to 1 or 2 or 3 patients, or not more than one at all. So this table can also be downloaded in a text delimited file or an Excel file. Next. So in the database, when I have one patient, I also have the choice of looking across the other 8,000 individuals that are there and see if there are other patients that overlap in terms of phenotype with my patient. So in that case here, we set the default to a feature match coefficient of 80%. So it's more or less an 80% overlap that I'm requiring to the database to tell me if the patient is a match or not to the patient that I'm working on. The other way to search for other patients that are similar to mine, next slide, is to go to the search tab and actually put the features that I'm interested. So you can see that here I put enchondromas and I put hemangiomas, and I have the option to say if the other individuals in the database have to match all these features or if they can match one or the other. So the next slide. And I told you that is a phenotypic database and a genomic database. So in there, I also have the CRAM files, the VCF files, and the ANOVA files of each individual that was sequenced. So I can also look across the entire database and look for patients that have not only this specific feature that I'm interested on, but that feature and mutation in a specific gene, or that feature and a specific variant that I'm interested on. I can make the search in each one of these, by any one of these feature keys, or group them in different ways. So only feature, only gene, only variant, feature and gene, feature and variant, gene and variant. So this is another way to find patients depending on what I'm interested in looking for. The next slide. And then from there, I can send patients to Variant Matcher and make it available to be queried by individuals from outside the project. So basically, every patient that was sequenced, in Variant Matcher, you are able to query the rare coding variants, not including the synonymous, in each one of the individuals. So if you find a variant in your patient with rhabdomyosarcoma and you want to know if any of the patients with all the disease that I sequence have the same variant, you can go to Variant Matcher and put the genomic position of your variant in there and the nucleotide change, and you can get a match if there is a patient, or we tell you that no, we don't have a patient with that variant. So the next slide, it's just going to show you that we also can select the genes that are candidate for each one of the patients, and from PhenoDB send them to Gene Matcher. So this is a way to share the data. Two ways that we have to share the data of the sequencing. And in both of them, we also share the phenotype information of the individuals that were sequenced when there is a match. So you can also know not only based on the gene or the variant, but also if there was a match on the phenotype. And of course, you can contact us to get more information about the phenotype if the information that you get was not enough to define the match or not the match. So the next slide, the last one, I think. Of course, part of this data, almost everything, not so much in terms of phenotype, but almost everything that we have sequenced through the kids first will be there in the DRC. And of course, if there's a way to put whatever else we have not sequenced through the DRC in there, we'll be also happy to do so. So this is all I have for my 10 minutes.
Thank you, Nara. Now we'll move on to Mary Marizia. Mary, I don't hear you yet if you're trying to.
Hello, how's that?
There you go. Hi.
Sorry about that. I had my headset turned off. Okay, so I'm the director of the Pittsburgh Center for Craniofacial and Dental Genetics, and Seth Weinberg is a co-director. And someday I'll see my team again, but here's a quick photo and the view from my office. Next. So the center has three major research themes: facial birth defects, facial variation, dental and oral diseases. Next. Next, please. And the facial birth defects study, which is, I'm sorry, go back. Well, it's okay. So there's a huge phenotyping component to our facial birth defect study. And in that regard, we've had multiple XO1s with GMKF from different ethnicities. Next. We're focusing on oral facial clefts in our GMKF studies, and these birth defects are seen around the world in about 1 in 700 live births and across all ethnicities, although there are some different epidemiological features by ethnicity. Next. So oral facial clefts have a number of subphenotypes: cleft lip with cleft palate, and cleft palate. Next. Which are grouped into cleft lip with or without cleft palate and cleft palate alone. We've done a lot of GWASs and now are of course embarking on whole genome sequencing. But what I will do in the next few minutes is try to highlight some of the things that are a little bit different from some of the other studies. And everybody spoke very well and were very inspiring about the ways they're organizing their data. So I won't try to cover again some of that ground, but we have various categories of what we think of as phenotypes that are covered in our studies. So we have the subphenotypes, and those are described in great detail for the probands. They can, again, be those individual subtypes. They can be bilateral, unilateral, they can be complete or incomplete, etc. And we have challenges in describing these because they're not, this level of detail is not reflected in the HPO or in ICD or any of the other common ontologies. So in a large project I've been part of for quite a while, the face-based project, there is a specific ontology for craniofacial development and malformations, and it covers human ontology and, interestingly, maps to zebrafish and mouse structures, which can be very useful as we do our genomic studies. So in addition to trying to define the subphenotypes, we also have what we call subclinical phenotypes, which I'll talk about in more detail. Then, as many of the other speakers have discussed, we have a large number of other types of information that we gather: pregnancy history, medical history, family history. And to adequately cover all of these issues, we have over the years accumulated large sample sizes, which in terms of GMKF includes almost 2,000 proband trios that have now been sequenced, with additional ones coming, again, once the COVID-19 situation has passed. Next. To get those large sample sizes, we've been collaborating with people around the world and within the US. So we now have a large number of case families, controls, and a large number of collaborations. Next. So in our deep phenotyping protocol, we have various questionnaires similar to what other people have talked about. Pregnancy history is very important for birth defects, demographics, personal and family medical history, developmental milestones. We do physical exams for details on the facial birth defects plus other physical features. An oral exam to get the dentition and the maxilla and mandible, limb, hands, digits. We'd get a speech sample by video for speech characteristics. And what I'll talk about a little bit more is some of the imaging that we do. So we take various types of imaging: 3D facial images, intraoral photographs, palate videos during speech, hand scans, ultrasounds, dental casts, upper and lower lip photos, etc. And we have all of those images in various types of file formats. But in addition, we derive summary variables from those images. Next. Next. So subclinical phenotypes, or what we call subclinical phenotypes, are physical features that are within the range of normal variation. So these are things that anybody could have. But one of our hypotheses has been that some of these features have increased prevalence in unaffected relatives versus controls, or in OFC cases versus controls. This is not a new hypothesis. So there's a lot of papers in the older literature looking at features such as fingerprints, handedness, and other characteristics in both cleft cases and their relatives, in part because there's a preponderance of unilateral cases, and among the unilaterals, there's a preponderance of cases where the left side is involved. So people hypothesize, well, maybe handedness is important, maybe fingerprints are important, etc. But we've gone beyond the types of things that were looked at in those early studies. So we hope that these can help us assist in heterogeneity delineation and in our genetic analyses, and more importantly, for some of the goals that are in the title of this workshop, being able to get to truly personalized genetic information. Because everybody has a different raft of subclinical phenotypes, subphenotypes, and other characteristics. Next. So one of the examples is looking at the orbicularis oris muscle using high resolution ultrasound. So that muscle is the muscle that circumnavigates the mouth, and we focus on the one that's above the upper lip. Next. So this is an ultrasound image of what is of the upper lip. You can see the yellow arrows pointing to the OO muscle, and it looks like a nice, dark, even band. So this is what we consider a normal OO muscle. Next. In contrast, the one below the muscle below has two areas that are circled where there are discontinuities seen in the orbicularis oris muscle. Now, in disordered development, which would lead to cleft lip, for example, this muscle would have actual complete discontinuities in the muscle and all the structures, the connective tissue and the skin, etc. But this is an ultrasound from someone with no visible defect, but their muscle has distinct discontinuities, right where we would expect the discontinuities to be if this was part of a cleft phenotype. So we keep these images, but we also rate each image as to whether there are discontinuities, whether the discontinuity is on the left or the right, etc. So that's all derived variables that are added to our data set. Next. In addition, as a particularly interesting subclinical phenotype, we've been looking at facial features in general. So we get 3D facial images of all of the individuals in our studies. This has become such an important part of our study that we actually have separate streams of analyses and recruitment looking specifically at normal facial variation. And the leaders of that study are Seth Weinberg and John Schaefer. But these are also assessed in our family members, probands, and controls for the OFC studies. Next. So the images that we take are 3D facial images that are done with photogrammetric methods, and they lead to a mesh of many thousands of points on the face, and you can define areas of the face or just really limitless numbers of phenotypes on the face using these images. This is one way of looking at those facial images that we are using to good effect in all of our studies, as recently reported in this Nature Genetics paper. So we start with the full face, the yellow face in the middle of this target. Then using clustering modules, statistical clustering, hierarchical clustering, we then go to each level, each ring around that center face. So the next ring leads to two additional phenotypes, the green nose area and the green everything else area, and so on out to the outside ring. So using all of these different characteristics, we then do genetic analyses of all of these characteristics, which we're doing by GWAS and also by whole genome sequencing. And then let's see, next one. So we've had good success in our normal facial development studies. For example, this is a locus that came up with very strong hits under GWAS with some of the facial modules. Next. So you see the circled modules here. So each of those three modules had very significant results with this locus. And if you look at the yellow indicators on those modules, it's focused on the forehead. Next. This is a heat map of one of the 3D image as a composite 3D image. So the red area again shows that the highest significance is found in the forehead area. Next. And Structural mutations in this locus lead to cousin syndrome, and obviously this has a very strong forehead phenotype. So again, these results came from basically normal faces, but it shows proof of principle for this method of defining phenotypes. Next. And just summarizing our Kids First studies next. So we'll continue with all of our themes. As part of GNKF, I'm really excited about this workshop and a lot of the discussions that have been going on about understanding what might be common genetic risks across all of our studies, and harmonizing our phenotypes will be very important for those endeavors. Next. Thank you, everybody.
So much. That was a great talk, just like everybody else today. Dr. Wise?
Hello.
Yeah. Oh, there you go. Hi.
Okay. I'm coming through now?
Yes.
Great. Well, thank you for allowing me to talk today. I will be very brief, and I will start off by saying I've already learned quite a bit. I'm inspired by some of the approaches that other people are taking. We are sort of at the other end of the spectrum. We are in search of phenotypes. This is our project. It revolves around adolescent idiopathic scoliosis. And so, you know, scoliosis, we talked about it earlier, a 3D spine deformity, common in children, and we're focused on the adolescent form. Being idiopathic is a diagnosis of exclusion. So that's why I say it's a phenotyping challenge. We're looking for those subphenotypes or subclinical phenotypes that are subtle and otherwise it might have been excluded. So again, the diagnosis here is excluding things like Marfan syndrome and neurofibromatosis and things like that. Next slide. AIS is genetically complex, which is probably not surprising, and most of the risk loci that have been identified so far have been identified by GWAS. And I'm going to talk a little bit about genetics because the stage we're at now is we are hoping that the genetics is going to point us toward those subclinical phenotypes or subphenotypes in this particular condition that will allow us to become more specific in the diagnosis. So the picture on the right is showing you a GWAS that was performed fairly recently. This was a large-scale GWAS that was organized through a consortium that we formed called the International Consortium for Spine Genetics Development and Disease. And so we had several fairly strong hits, and a GO gene ontological analysis pointed us to cartilage development. And particularly, these genes are pointing us to the cartilage, the so-called matrosome of cartilage, the extracellular matrix. And so we think this is beginning to tell us that what is perturbed, or at least part of the underlying risk, is probably at the level of the intervertebral disc and possibly the growth plate of the vertebrae themselves. And that's what the cartoon on the right, part D, is showing, is just a cartoon of some of the molecules that we have identified that carry risk variants for AIS. So that's one take home here, is that just very recently, our data is pointing us to the cartilage matrosome. And the second point that isn't illustrated here, but that I will tell you, is that the alleles that we are identifying by sequencing, I talked about GWAS now, but by sequencing are pointing us to hypomorphic mutations, which might not be too surprising. We're not finding those strong, obvious loss of function mutations. And this is also what's coming out of a forward screen in zebrafish that's part of being led by Lila Solnikow-Kretzel at WashU as part of our program project funded by NICHD. So these hypomorphic alleles, some of which may reside in genes for Mendelian disorders. Next slide, please. So with that knowledge, we elected to perform a focused EWAS, exome-focused genome-wide analysis specifically looking at the cartilage or the matrosome. And what was identified, one of the factors that we identified, I'm showing you in this slide, this is unpublished work, we have association in a SNP in collagen 11A1. This made sense to us. Our previous study, led by Chris Gurnett, that we participated in, has identified rare variants in collagen 11A2 in some cases of AIS. And so, and part B is showing you replication through our international consortium. So we're happy that this is pointing us to new biology. Collagen 11 is a minor but important component of the intervertebral disc. It's a fibrous collagen. But the other interesting part of this is that the collagen 11's Mendelian mutations cause Stickler syndrome. And Stickler syndrome, it has several hallmark features. So we're asking ourselves, do our patients that carry these variants have any subtle features of Stickler syndrome, hearing issues, visual issues? I can tell you that they certainly have no overt Stickler syndrome, but that's where we're using the genetics to go back and look through our data on all of our patients to see if there are subtle features that would distinguish this subgroup of patients. The other interesting note is that our associated SNP is part of an LD block that it has previously associated with lumbar disc herniation. This was by Shiro Ikagawa several years ago. So we're also going back and reevaluating our patients to see if there's any lumbar morphology that might be interesting. We have about, in our cohorts total, we have about 2,500 probands. Not all of those were included in this analysis, but that is the size of our data set right now. And for all of those people, we have a fairly straightforward health history. And we also have access to their medical information in an EPIC platform here at Scottish Rite Hospital. And this is a platform that was custom designed for research, so we can pull data as discrete variables. And we also have pretty granular information in terms of imaging on all of these patients that we can access. And we also have the ability to recall patients if they gave their consent. So this is an example of where we are right now. Our activity is going back and reviewing the phenotypic information for more subtle features and potential subtle features in these patients. And again, I appreciate the conversation before on how we can sort of better organize ourselves to do this. Next slide. I was trying to forward the slide myself. And then I wanted to turn just briefly to one of our Gabriella Miller Kids First projects. So through GMKF, we very happily have performed whole genome sequencing in families that show dominant forms of AIS. And I have to say, two of the example pedigrees are shown on this slide. We say dominant like because there are still issues of some complexity and reduced penetrance and so on and so forth. But families suggest to us that there are genes with conveying stronger risk in these families. So in our first round of sequencing in a subset of these families, we identified mutations in the gene called HAPLN1 in several families. And burden tests approached significance, and there was an increased odds ratio for this gene. The reason that was number one, and number two, the gene encodes cartilage link protein, which is a part of that cartilage matrosome that we were talking about earlier. And we know that it is regulated by another well-known AIS risk gene, GPR126, and it's dysregulated in a knockout of that in the mouse. So we have focused on this as an interesting candidate for AIS. And we're bearing in mind that this gene is also expressed in brain and heart. And on the right, you're seeing the pedigrees where we saw co-segregation of one is a novel mutation and the other is a very rare mutation. both of which occur in the B prime link domain, if you see in Part B. And we identified a couple of other mutations that I'm not showing you here, also in that B prime link domain. And modeling suggests that this would be deleterious to the protein. This protein is important in maintaining the hydration of the intervertebral disc. So we went back and reviewed these families, and in one family with the G336 mutation, the family reported pretty early onset lumbar disc disease. But by the early 20s, individuals in this family were reporting disc disease. In a second family, the proband had mitral valve prolapse, and she presented with marfanoid features, but none of the testing nor sequencing has identified any mutations or any sequelae that she actually has Marfan syndrome. But interestingly, her father died of aortic dissection. So again, this is interesting features that we're now going back to look more carefully for these potential associated phenotypes throughout our database. But then, of course, we'd like to test causality and test the consequences of these mutations. And so, thankfully, partnering again with GMKF and the COMP2 initiative, these mice are, we've made one of the point mutations, and a couple of more of those are in progress with the UC Davis group, and we really appreciate that. And once we get through this COVID thing, we're very excited to move forward with these mouse studies. Sorry, next slide, please. So that was just to show you that we are looking for more phenotypes, and we're allowing the genetics to point us toward those potential subclinical phenotypes in our patients. So lastly, I wanted to mention an effort that we are hoping to kick off, and that is utilizing the information that we have access to here. Here, most patients are very aggressively braced as soon as they present with more than about a 20-degree curve, because there has been evidence that bracing can correct AIS if it's warm, and it has to be compliance with bracing is tough. So here at Scottish Rite, compliance is monitored. Every patient who is treated wears a brace with a sensor, and all of that data is in the medical record and is accessible. So it's very, very granular, the amount of bracing over time. And so it occurred to us that that gives us the opportunity to identify true treatment non-responders, people who wear a brace faithfully and still have this really aggressive, progressive disease that sends them on to surgery. And so we are collecting those patients with the goal of performing genetics to understand the variants that are driving a truly aggressive spinal deformity. Next slide, please. That's it. That's it for me. Just to summarize again, we are in early stages. We are searching for phenotypes, more subtle phenotypes that go along that can help us subgroup this disorder and lose the name idiopathic eventually. And again, I appreciate what I've learned today in terms of how we can start collecting phenotypic data in using nomenclature and language that would be consistent with what other people are doing. Thank you very much.
Thank you, Carol. Sarah Rabel from Ian Kranz's group. Sarah, I can't hear you yet. If you're
sorry, can you hear me now?
Yes.
Okay, great. All right. So our EXO-1 project is focused on genomic diagnostics in Cornelia de Lange syndrome, related diagnoses, and structural birth defects. And just to very briefly go over CDLS, CDLS is a pretty rare diagnosis. It's about 1 in 10,000. Most cases are sporadic, though we do see some rare familial recurrences. Most are, or all are due to dominant inheritance. due to de novo mutations, and there can be a lot of variable expressivity. The mutations are in cohesin, structural, and regulatory proteins, and for this diagnosis, it's pretty genetically heterogeneous, and there's 5 main genes that are the causes of kind of typical or classic CDLS, but about 30% of patients with a CDLS diagnosis don't have a known underlying genetic etiology, and we think there are many reasons that can be contributing to that. Some may be mosaic, which is something that's been reported more recently in the literature over the last few years. And then also over the last few years, there's with exome sequencing, there's been new gene discovery of these kind of CDLS-like genes, such as BRD4, ANKRD11, and a few others, which we think make up a portion of this 30%. So next slide, please. Okay, and this is just a picture of the cohesin ring. So cohesin is a multi-protein ring complex, and you can see at the bottom left that NIPBL is the most common gene that causes CDLS. About 60% of patients will have a mutation in this gene. All individuals that have more of a severe classic presentation with limb defects typically have mutations in this gene that are truncating, but we can see more milder presentations in NIPBL with missense mutations. And there's been a lot of work that's been done over the years for a lot of these other genes to kind of parse out genotype-phenotype correlation among these. Next slide, please. Okay, so this slide really has 2 main points to this. First, to obviously show how CDLS is a multisystemic diagnosis with such a wide range of clinical features that are affecting almost every body system. And also, one of the reasons why our team was so interested and excited to study this diagnosis is because since every system can be involved, whatever genes were found to cause CDLS, we believe we're going to be key developers. And we really plan to use that information found from Mendelian disorders that may be easier to map to help get insights into the cause of some of these individual isolated birth defects. Next slide. Okay, so this is the first of a kind of like a 3-slide part series of our approach, and it's really meant to show kind of the basic concept for how we use these rare developmental disorders like CDLS to get insight into isolated birth defects. And so when we think of a syndrome, we think of a mutation in a particular gene that's critical for development and differentiation of a bunch of different cells and organs. So if you have a mutation in a gene, like in this picture, that's important for the heart, brain, muscle, and other cells, where that gene is directly active, will cause differences in all those tissues, and you'll end up with a syndrome. So like for this example here for Alagille syndrome, when you have a mutation in JAG1 or Notch2, which has a direct effect on certain cell subtypes, you get liver, heart, and facial involvement, and which results in Alagille. So when there's a mutation in a gene that's important in the development of many organs and tissues, you will always get a syndrome. Sometimes it can be subtle, like in the case of Alagille, if you initially think it's an isolated heart defect, but if you look closely enough, you may find some subtle facial features or some elevated liver enzymes, and it's not just truly isolated. And a lot of people have put a lot of effort and time into looking at these types of syndromic genes and looking for causes of isolated birth defects or looking for the cause of isolated heart defects in the example of Alagille syndrome. And the yield has been pretty low. The next slide, please. So this is where we think really kind of like the money is for isolated birth defects. So where we have a mutation where the lightning bolt is in a regulatory element that controls that gene in a specific tissue that's developing like the heart in this image. So even though that gene itself is expressed in all these tissues, and if you have a mutation in that gene, you would get a syndrome, If you get a mutation in the regulatory element that only affects that gene in those tissues, then you'll get a real isolated birth defect. So the example here is for limb defects. So there was a group that found a point mutation in a regulatory element that just affected expression in sonic hedgehog in the limb, and they got isolated limb defects, which is not something we typically see with sonic hedgehog. When you have a mutation in that gene, you get holoprosencephaly and some other features as well. Next slide. Okay, so this is what we see in CDLS. So this is where we have a mutation in a master regulator that controls the way these regulatory elements interact with their genes globally. So when we have this master switch, we can hopefully use that to identify these downstream targets, what these downstream targets are, and which of these regulatory elements of gene interactions are disrupted, and try and find those downstream genes that are causing the individual birth defect. So this in CDLS causes the syndrome, and cohesion facilitates interaction of those long-range regulatory elements. And so we're hoping that once we can kind of map all of this out, we'll find some key genes that are dysregulated. And then we can identify what the regulatory elements are that are controlling those genes. So like, for example, if we find a gene that's important in, let's say, diaphragm development that's dysregulated, we can find what is the regulatory element that regulates that gene in the diaphragm that's being dysregulated because cohesion isn't working properly. And once we do that, we can look for mutations in the regulatory element as a cause of isolated congenital diaphragmatic hernias, which kind of gets back to that previous slide, where we think that the mutations are going to be in the elements that regulate these genes in specific tissues, because few genes are needed in just one tissue. So overall, we're just really hoping that these disorders that are master regulators of gene expression, like CDLS, will give insight into understanding the genetics of isolated birth defects and or of those birth defects that are seen in constellation in those syndromes. Next slide. Okay, so that's kind of just the background that I wanted to give you about why we think it's important and valuable to study CDLS, and to get more into our phenotyping efforts. So our cohort at CHOP, we've had an ongoing research study for CDLS for over 25 years now, and we have a pretty robust cohort of close to 3,000 individuals. And I think what's important to take into account, especially with regards to phenotyping, is that a lot of individuals that have been referred for our research program do come from outside referrals. We are a large referral center. We have a robust clinic that we hold, so we do see a lot of families in-house, but we do have a lot of collaborators and a lot of other researchers who have sent us samples and information over the years. So there certainly are limitations to that, which we will discuss in further slides. But for our cohort, specifically for our Gabriella Miller project, 400 genomes were being sequenced. We have 178 probands, 96 of those are trios, 30 are duos, and 52 are singletons. Next slide, please. Okay, so where we kind of started with phenotyping for our cohort was that we had an existing FileMaker database, which is something that was created many years ago. And as we started going through our cases and pulling phenotypic information, we realized there were quite a few limitations we were running into. First, obviously, for a lot of those patients that were referred to us from outside institutions, we were limited based off of the detail of the medical records that were sent to us. And even within our FileMaker database, the phenotypic fields that were in there for capturing the clinical history, we felt needed to be expanded, as with time we've learned more about the phenotypic spectrum of CDLS, and there were some variables that we felt weren't being captured. We also were finding incomplete data entry, and the big kicker is that HPO terms haven't been historically captured in this. So for some time, we had been talking about doing kind of an overhaul and creating a new database for our study, and this kind of propelled us forward with that. So on the next slide, I have here, we did establish a new database. It's a database that's on a MySQL platform, which allows for either sharing and transferring of data for future collaborations as well. And on the next slide, so, and how we started with characterizing HPO terms, there's not an HPO term for CDLS, but the orphanet term for CDLS did have 100 annotated HPO terms already associated with this diagnosis. So we felt that was a good place to start in terms of standardizing this process. So we pulled all of those HPO terms and they were put into our database. And on the next slide, so we had, what we started with is we had a team of our clinical researchers, genetic counselors, and physicians who have vast experience in caring for children with CDLS. We're reviewing medical records, paper charts, our old FileMaker database, and selecting which of these HPO terms were appropriate for each of these cases. However, as we started going through this process, we were finding that even though we thought this would be a good way to kind of standardize this, there were certainly some issues that we were running into. So first, we were finding that out of those 100 HPO terms, this was certainly not inclusive of the full phenotypic spectrum, as it wasn't really it wasn't allowing us to capture some of the more mild cases or the phenotypes of these more kind of CDLS-like or atypical or overlapping cases as well. So an example I have here is that there's the term HPO, the HPO term intellectual disability severe, but it didn't allow us to capture for moderate or mild intellectual disability. So those had to be added. Again, common features of CDLS surprisingly were not captured in those 100 terms. So for example, even though speech delay was in one of those first 100 terms, developmental delay or motor delay was not, and different behavioral phenotypes, which can be very common in CDLS, was also not captured as well. And then on the next slide, there's just two more limitations that we were running into. So we were also finding, as other people have mentioned, this and is an issue with HPO terms is some things were just certainly too specific or not specific enough. And I have a few examples here. I won't run through all of them, but one of the examples is that for heart defects, which can be common in CDLS, the two most common, certainly ASDs and VSDs, but there are other heart defects that were not being captured. So we had to include more general terms or other more specific terms to make sure that we are getting a complete picture of and capturing the complete phenotype. And then lastly, for some of our CDLS atypical types of cases and that had maybe some pretty unique or uncommon features, they obviously were included in those 100 HPO terms as well. So we kind of took this on a very case-by-case basis, and we're finding that almost with every other case, we're maybe adding another HPO term to our new database in order to accurately capture as much as we could. And the next slide. So some additional considerations for that we ran into during our phenotypic experiment was that with facial features, which can be pretty distinct in CDLS, but especially for some of the more mild cases, there can be a lot of variability. And we know that there can be subjectivity to this, depending on which clinician is performing the physical exam and documenting those facial features. Also, again, for our outside referrals, we were limited on the types of photos that were sent to us. But we tried to account for this and standardize this as best we could by having clinicians in our center with experience caring for and managing patients with CDLS review all of those cases and have their input. And then
Sarah,
lastly,
could you
just for ethnicity.
we have to move on.
Oh, sure.
Is now a good stopping
You
point?
said
Sorry.
I'm sorry, what did you say?
Is it okay if we stop there? Did you get through the meat of your presentation? Because we're running out of time.
Yeah, that's fine.
Okay, I'm sorry about that, but we really have to move on. I think that we're actually going to go ahead and skip our next break because we simply don't have time for it. So we're just going to move on to Allison Heath's presentation.
Okay, thanks, Valerie. I think we have basically 15 minutes left. As Nicole's advancing through the slides, there is a bunch of summary information about the survey we sent out that some of you might find interesting. I'm going to go through this really quick so we can hopefully at least have a few minutes of discussion, which we had reserved originally 3 minutes to do. Many of you have seen this. This is really kind of the exon 1 data path, and we've been focusing on the phenotype data today. If we go to the next slide. So many of you have interacted now with this application we call the data tracker, really recognizing that when the DRC started, we had a huge retrospective work behind us, getting data, recognizing that there wasn't a lot of standards when the program first launched, and really thinking about, how we arrived here today. How can we do this better? What can we do prospectively? So I wanted just to give a little peek under the hood today, because this is where you start if you hit next, Nicole, I think it should. But what happens next? I think many of you, this is kind of the endpoint that you give us the data and then you get your genomic data. And I just want to talk a little bit about the process. It gives some insights into, I think we have a really good team and platform now. We really want to think about how can we expand this in the future. Today's talks have just been really inspirational and inspiring of, what could we really bring to bear together with the phenotypic and genomic data. Next slide. So as many of you know, and I think there's a high variability across what people were able to provide, really depending on whether you had created data management systems before, whether this was the first time kind of working with a program like this. And we really worked hard to try to do the data justice, right? I think a lot of us have worked very hard to collect this, to do these studies, and even though it might not be in the best format, we really want to get it to a better format. So the team here has created a lot of ingestion libraries, a lot of ways of taking this non-standard data and having clean map join data, and really worked hard to get to that point, and both preserving the original data as well as the curation of ontologies that are Nicole and Deanne and others have really worked hard for. So at the end of the day, we take that data in there, we go through these ingest libraries, we create something called ingest packages that are automated and allow us to rerun as we get new data in. We email a lot of you and ask for fixes, and we've really tried to smooth that process out on our side, that as soon as new data arrives, we try to turn around pretty quickly. From here, if we go to the next slide. As I was saying, we do a lot of this hand curation right now, and I was hoping we could talk more about how we could do this better, but we'll see what we have with time. Next. Just some quick stats. I mean, we're up to 25 studies. I think the question I asked myself is, how would you do this if there was 250 studies? How do you do this if this was 2,000 studies? How do we scale this? How do we have other people involved and really think about how we can grow these efforts? The next. For those kind of more techie oriented, the target of all that work that the data wrangling team does is the centralized database that helps us organize and do this. And I think if you go to the next slide, Nicole, we have recognized that there are many limits. Oh, I skipped this one. Next one. There are many limits to what we did originally. When we first got started, we just wanted to bootstrap, get things in there, start centralizing it. We did do, I think, a good job of choosing initial ontologies to use, but there's always new terminologies coming online. There's disease-specific things we'd like to support. And, we would really like to be able to expand that. And then, I think there's a lot of these kind of cause and effect, and, how do people indicate how the data was captured? Is it really just a series of time points? Do you know that different things are tied together? And, I think some of the hope was to really gather some of these analysis questions. I think today got us a lot of background and, was looking forward to this brainstorming session. But, I think hopefully we can continue doing this in the future. But at least internally at the DRC, we've been asking ourselves these questions a lot. And over the last 6 months, we've really moved towards creating a new clinical data platform. So that's the next slide. And really thinking about, right now we're kind of in this spreadsheets, people exporting databases, kind of the dbGaP phenotype space, which I think is a great place to start. We hear a lot of people using REDCap. We hear a lot of people are looking at EHRs. And so we were really thinking about how do we create ways that we can have this data integrated and flow, and also, give you a view back on your data as we go through this process. I think the idea of it kind of being handed over and not knowing what's going on, I think creating transparency through this process is really what we want to do in this next phase. And you heard Melissa talk about Fyler a little bit earlier, and I think we've really found that it's a good way to kind of capture the raw data. So it allows us to bring in what might seem like disparate information from structural birth defects and cancer, but since it was really meant for like the full medical record system, there's a lot of flexibility that we've been working with technically to bring that in so that we can view this, and then we can start creating these applications that can look at our data, hopefully interoperate. We're heavily involved in the NIH Cloud Platform interoperability work, and really thinking about how can we cross-intersect and create tools that can look not only over Kids First data, but other data sources coming up. So, these are some of the things we were doing. And, the next slide, I really do think data is a team effort. So here are your two of your original ingest library developers, Natasha and Avi. I think probably wondering if that space or that data would really be there. I think this is a lot of what we do, and I think it needs to be informed by people who are experts in the diseases. We can make guesses, assumptions, look at things that are strange, but we aren't at the experts at the end of the day. So we've been thinking about how do we incorporate that and really create a life cycle ecosystem of this. And I think really the message here is everyone is really part of this team. We all work in our own areas, we have our own expertise, but I think figuring out how we come together, how we create platforms that enable this. Now that we've kind of gotten over the initial hump of going through all the retrospective data and kind of getting some of the operations done, this is a really good point to start thinking about what the future looks like. Hopefully I went through that quick. Thank you.
Thanks, Allison. I think if we could actually skip over James's component and go right into the brainstorming session. Melissa, do you want to take that over?
Yeah, so if you guys can just navigate to that link, it's also in the chat, and we can paste it there again. So the idea with the dot storming, and I guess especially given that we have very little time, I've kind of seeded it with some ideas from today's discussion, and I see that some others have put some ideas in there. I'd really like to continue to populate these ideas. These are essentially like, what are the core challenges that we as a group would like to try to address to better create and better use interoperable phenotype data for and by the Kids First portal and the Monarch initiative. And so we really need your help. And I think maybe what we could do is over the next few weeks, while it's still fresh in your mind, kind of populate and comment some of these ideas. And then there's voting, and you can vote on the things that are most important to you, as I see somebody is doing that turns green. When you vote, you get 3 votes. But we really need some more ideas on the board here. And then what will happen is eventually we'll have a face-to-face meeting, and we can actually do some preliminary design work on the things that people want the most, and then bring those to you and kind of iterate on designs and plans and strategies. One thing I did notice as we were going through all the talks, and I thank all the speakers for taking the time to put together those slides, what an incredible diversity of phenotype data and modalities. It's just the common need for some tools that are configurable. So how many different databases did we see today where just simply capturing phenotypic information is laborious and heterogeneous across all those different sources? If we had some common tooling and you could display it however you wanted in your own systems, but knowing that it was sort of being generated in a born interoperable manner that might be helpful. Similarly, like when terms in the ontologies are too granular or not granular enough or missing, what should you do about that? And how can we help users, put information in that is still useful even in the absence of the perfect match? So just those kinds of commonalities where for our teams to collectively build you all the best possible tools and resources for helping you with encoding your phenotype data, just trying to brainstorm. And I'm just going to open the floor for anybody who wants to make some comments because I've done plenty of talking already. In all of 5 minutes. If anybody wants to just like lay it out there with your favorite problem challenge that you'd like to see solved.
Hey, this is Brian from OHSU. Hey, Melissa. Hey, everybody.
Hey.
The two categories of normalization or harmonization that I think is worth calling out separately, or that I'd like to throw out for consideration, are drug/treatment and to a lesser extent tissue location or specimen characteristics. The HPO and Mondo terms and tooling around that are great for drilling down into disease/phenotype, et cetera. And Fyler, as much as I've been catching up to speed with it, is great for describing the structure of observations and treatments, et cetera. But at the end of the day, we still have spreadsheets and external databases that need to be processed. And terms that are used for drugs, tissues, variants, disease progression, things like that. All of those maybe not be as all-encompassing a problem as phenotype identification, but in order to get a complete picture of a study, at least those three concepts need to be harmonized to the same degree of rigor. Does that make sense?
Yeah, and I think, part of it, is that there are such things that exist. I mean, we actually, have, a library of drug identifiers. And we even have some preliminary modeling. This is actually one of our GA4GH conversations that maybe Peter can speak to, about how you model treatments in relationship to phenotypes, especially where temporality is concerned. Mm-hmm. But part of the problem is, like, what could Kids First do to help submitters encode that in the same way? So often what you see is you'll see just a list. You don't actually see the relationship between the treatment and any given phenotype. And you also don't usually see the temporality and how the phenotype change in response to the treatment. And then you don't see any identifiers almost always. So that's, I think, maybe where part of it is just best practices. Like, could we come up with just sort of recommendations, even in lieu of having any kind of standardized CRF form elements, but just simply like, if you want to use identifiers for drugs, use these ones, and here's how, kind of thing. It seems like part of what you're asking for is that.
Yeah,
histopathology-wise, yeah. Sorry, go ahead.
I was going to say, in lieu of is the use case I'm thinking about, where there is no relationship with the submitter, in the sense that, the data exists, the submitter is long gone, the data set needs to be incorporated into some aggregation or some search engine or whatnot. And there's no opportunity to go back and say, please use these terms or please implement the standard. It's up to the consumer of that data to harmonize it.
Here's Peter. Could I just make a short plug?
Sure, yeah.
So thanks, Melissa, for mentioning that, but the GA4GH is in the process of considering an extension to the Phenopacket that will capture treatment history essentially, and in the course of time. Part of the motivation for this is to capture also the cancer phenotype. And so we're just starting this project. Would love to have feedback, collaboration, beta testers, and so if anybody here is interested, please get in contact with myself or with Melissa, or just go to the GA4GH clinical pheno work group. But I'd be happy to point you in the right direction.
Great, thank you.
Thanks, Peter. I think that's a great plug to have to close. You know, and I think some of us were hoping to convene in person for this meeting and talk a little bit more about those resources and efforts that are ongoing already. You know, if this thing goes on longer, maybe this thing is going to become a webinar series where we can maybe learn a little bit more about those types of resources and discussions that are happening elsewhere. Because I'm not sure, you know, everyone in this group is super familiar with GA4GH and phenopackets and the things that are out there.
And to confirm, Melissa, you're planning on keeping this dot starming page open, right, for people even once the call ends?
Yes, we will keep this open. I got a free version of it for 30 days. So maybe, what do you all think about having maybe a regular cycle of these kinds of calls? I don't know, maybe once a month, where we can actually go over some of these priorities and make some project plans. And then that would also feed into a face-to-face as soon as we're able to all actually get together in person sometime this fall.
I think this is Adam. I think it would be a wonderful idea to take some of this and then iterate on it as sort of a working group with real data and real use cases. You know, it would benefit the DRC tremendously.
Yeah, so I would love to see maybe like a monthly, hourly call, and then also maybe we could follow up with some folks. Maybe as a follow-up, we'll send an email out to everybody, and please do keep populating this. I think it's really helpful for all of us to kind of see what the best focus is for our efforts. So I know we're past time. I don't know if Adam or Valerie or Nicole would like to close this out.
We'll let Valerie and Nicole.
Yeah, go ahead.
From my perspective, are awesome. No, I just saying thank you, and then Valerie or Nicole, please close us out.
Yeah, thanks everyone for coming. We'll share the recording with everybody, and yeah, we'll follow up with potentially having a monthly meeting and other plans for the future to keep on working on these topics. And these are really great talks, and really appreciate everyone joining us today.
Thank you.
Thank you, everyone.
Thanks.
Bye-bye. Be safe, everybody.
Bye. Thank you.
Bye-bye.